Hello and good evening, and welcome to the second day of the third Philippine Herbal Medicine Summit. Yesterday, we kicked off the summit with a welcome address from Dr. Cutionco de la Paz, a keynote from Dr. Domingo of the FDA, with plenary sessions on initiatives to improving standards for herbal medicines, and updates on herbal therapies for COVID-19. Tonight, we will have the stakeholders meeting, areas for growth and cooperation, followed by the Plemix Herbal Medicine Research Contest. Some general reminders to all our participants. Please use your first name and last name as your username for easy identification. Please ensure that your microphone is on mute and your web camera is turned off throughout the duration of the summit. Questions will be entertained at the end of each plenary session. Please use the raise hand sidebar function if you have any questions or simply type at the Q&A chat box. Please monitor the chat box for any announcements. And now we will begin our first plenary symposium for tonight. Our stakeholders meeting, areas for growth and cooperation. We have Evangeline C. Amor on the researcher's perspective. Professor Amor is the current Assistant Vice President for Academic Affairs, Curriculum and Instruction of UP Diliman. She is a professor at the Institute of Chemistry of the College of Science, specializing in natural products and organic chemistry. Her research interests include isolation and structure elucidation of bioactive, such as analgesic, anti-inflammatory, hypoglycemic, and anti-cancer compounds from Philippine plants. Structure activity relationship studies, mutagenicity and plastogenicity studies on medicinal plant preparations, and enzyme inhibition studies. She earned her Doctor of Philosophy, Master of Science, and Bachelor's degrees in chemistry from UP Diliman in 2003, 1998, and 1989, respectively. She did a research fellowship at the Center for Advanced Sensor Technology, Chemical and Biochemical Engineering Department, University of Maryland at Baltimore County, Maryland, United States, from September to December 2006, funded by the Organization for the Prohibition of Chemical Weapons. She was recognized as UP Scientist 1 from 2012 to 2014 and awarded the National Research Council of the Philippines with the Outstanding Lifetime Achievement Award in 2017.
Good evening to our distinguished panelists and guests on this second day of the third Philippine Herbal Medicine Summit. Ensuring quality of herbal medicine through research, regulation, and cooperation is a timely theme as it provides the platform to further strengthen academe-government industry linkage as translational research and innovation become buzzwords in the emergent fourth industrial revolution that we find ourselves in. The use of herbal medicinal products and supplements have increased in the past decades, and these make an enormous contribution in the primary healthcare of developing and least developed countries. It is therefore imperative to ensure their quality, which determines to a large extent their safety and efficacy. Allow me to share my perspective as a researcher with respect to ensuring quality of herbal medicines by making use of the DOST Tuklas Lunas program framework. Tuklas Lunas is the Philippines Drug Discovery and Development Program that, in quote, envisions to produce world-class medicines derived from Philippine biodiversity, leveraging on local expertise, end quote. The framework shows two tracks, the herbal track and the drug track. The herbal track yields standardized herbal supplements and/or standardized herbal drugs. The drug track yields synthetic drugs. The first step is sample collection. Once the plant to be collected is identified, whether from biodiversity information or mined from the scientific literature. At this point, quality is ensured by making sure of the reliability of the source of the plant sample. This was covered in yesterday's session. The importance of the quality of source material is dependent not only on intrinsic or genetic factors, but also on extrinsic factors like environmental conditions, good agricultural and good collection practices, for medicinal plants, including plant selection and cultivation. The second step is extraction or expression that yields extracts for bioactivity and toxicity testing. This generates non-toxic bioactive extracts that are then standardized. As the plant sample is developed into an herbal supplement after preclinical studies or into an herbal drug after clinical studies. Standardization in this process contributes to the quality control of finished herbal medicinal products. Quality specification and standards for the herbal materials are established. The non-toxic bioactive extracts feed into the drug track, where isolation and structure elucidation of active constituents are done in order to generate drug candidates. These then go into pharmacokinetic studies, absorption, distribution, metabolism, elimination, and toxicity, prior to preclinical studies and clinical studies to generate synthetic drugs. The Tuklas Lunas Protocols for Drug Discovery and Development Manuals, published by the DOST-PCHRD, provides guidance in the processes from sampling to preclinical studies in the Tuklas Lunas program framework. Adoption of this protocol allows for comparability of research results generated by the Tuklas Lunas research program. To date, the program has supported and partnered with 27 institutions nationwide for the implementation of projects addressing various stages of drug discovery and development. The following factors have been identified to influence the quality of herbal products. Herbal products are mixtures of many constituents, and the active principles, in most cases, are unknown. Isolation and identification of these remain to be challenging because of low yields. In instances when reference compounds have been identified, these may not be available commercially. However, advances in analytical methods have helped address these factors somehow. For example, the LC-MS-MS method is used in identifying putative constituents of an extract. Standardization sometimes requires selective analytical methods, and adoption by the industry becomes challenging in terms of cost and personal expertise. For example, the use of NMR in quality control of herbal products. This may be an area where academia and industry can cooperate. Reproducibility of biological activity of herbal extracts are affected by the characteristic of plant materials being chemically and naturally variable, the existence of chemo varieties and chemo cultivars, and the variability of source and quality of the raw material. This is why there should be proper standardization and quality control of both the raw material and the herbal preparations. Botanical extracts made directly from crude plant material have been reported to show substantial variation in composition, quality, and therapeutic effects. Thus, it is recommended to have standardized extracts, which are deemed of high quality if it contains consistent levels of specified compounds and subjected to rigorous quality controls during all phases of the growing, harvesting, and manufacturing processes. And because the quality of source material is critical, promotion of mediculture is encouraged. This is the cultivation of medicinal plants on a scientific basis. Emphasis is on genetic stability and uniformity of plant population in order to get reproducible results. The sustainability aspect may also be addressed by mediculture. At the same time, interest of medicinal plants production actors should also be taken into account. This includes the enforcement of relevant laws and the implementation of benefit and knowledge sharing. This is an area where academe, government, and industry can cooperate. Areas of growth may be in the following considerations where much research still needs to be done. Herb drug interactions, synergistic action of unknown constituents in the herbal preparations, production of active secondary metabolites after intake of the herbal preparations, and in animal healthcare. Research on medicinal plants in the Philippines have gone a long way from the time that I started as a research assistant working on natural products in 1990. The funding support of the DOST and the DOST PCHRD is gratefully acknowledged, as this significantly contributed to research capacity building of higher education institutions. Acquisition of equipment and materials for implementation of research projects on medicinal plants were enabled. Quality medicines leveraging on our biodiversity will be a reality with the concerted efforts and cooperation of academe, industry, and government. With this coming to fruition, I look forward to the integration of herbal medicine into our national healthcare system. Thank you for this opportunity to share my perspectives, and good evening once more.
Okay, thank you, Dr. Amor. We will now have Dr. Benito Atienza of the Physician or Medical Practitioner's Perspective. Dr. Atienza is the current president of the Philippine Medical Association. He completed his degree in medical technology at the University of Santo Tomas and later on pursued medicine at De La Salle Health Sciences Institute College of Medicine. He is the first Filipino doctor to be awarded as one of the caring physician of the world by the World Medical Association during the WMA convention at Santiago, Chile in 2006. The first recipient of the PMA Dr. Jose Rizal Memorial Award of Distinction in Community Leadership and the youngest awardee of the Outstanding Filipino Physician in 2009. He was also awarded the PPS Dr. Fedel Mundo Award for Community Pediatrics in 2000 and the Most Outstanding Community Pediatrician by the Community Pediatric Society of the Philippines.
Good day from the Philippine Medical Association National Officers and Governors. I am grateful for the privilege to be able to share my insights and perspective to all the stakeholders present in this Congress about the relevant topic on herbal medicine. The pandemic caught us in the midst of our busy world, and now we are living in the SARS-CoV-2 and its variant in our midst. Its impact hitting the many facets of our lives, we all learn new things. We acquire new knowledge, we adjust and adapt, and we innovate. We became resilient. We also learn to look back and start practicing the basic things we have forgotten about. We search the answers to challenging questions, and we challenge our thoughts through research that paves way to discoveries, objectives, and factual. Herbal medicine was defined in the World Health Organization General Guidelines and Methodologies on Research and Evaluation of Traditional Medicine, including herbs, herbal materials, herbal preparation, and finished products that contain active ingredients, parts of the plants, or other plant materials or combinations. The use of herbal medicine has been a part of the traditional, complementary, integrative, and now better known as the TNCM. Its popularity has increased over the years as people shift choices from artificial to natural or synthetic to organic. As physicians, we are sometimes confronted by situations where our patients will ask about this very topic. Questions like, are herbal medicines effective? Can we replace our medicines with the organic or plant-based remedies? Can we take herbal as supplements? Are not new to us. In some occasions, patients even bring with them a particular herbal medicine and request that the Chinese characters in the product label to be explained. Without so much knowledge on traditional medicine, such scenarios can be disappointingly awkward. Are herbal medicines effective? According to World Health Organization Bulletin, herbal medicines are getting significant attention in global health debates. In China, traditional herbal medicine played a prominent role in respiratory syndrome SARS, and locally we have quite number of them which can alleviate myriads of illnesses endorsed by the Department of Health. Those that underwent extensive studies and were proven safe and effective are currently put to use. As physicians, we are bound by certain ethical principles. Our decisions are of scientific foundation. We believe in evidence-based medicine, and we adhere to the medical standards that put our patient safety to the highest level. If there are objective evidences of the herbal products' efficacy and safety profile, there is no reason why such can be withheld from our patients. With that, we call on for more research in order to generate more data that can provide us the scientific basis for its use. Not all physicians are trained to practice TNCM. Most of us are aligned to Western medicine. The use of herbal medicines is really questionable to those who do not have open mind and clear view in this particular field of study. Practice with lack of expertise in traditional and complementary medicine can be detrimental to other people's health. With that, we call on for education and proper training of those whose heart reaches out to this kind of endeavor. There are also concerns on the use of herbal products with components that can make them targets of abuse and misuse. We have learned of certain herbal medicines in the past that were used for the purpose other than what they are intended and indicated for. In this regard, we see the importance of the control of the regulation when it comes to herbal medicine that had potential to be used unscrupulously, posing hazard to the health of our user. We believe on the collaborative efforts of our government agencies such as the Department of Health, PDEA, and the FDA Philippines in laying the ground for appropriate mechanisms and strengthening further the control, the regulation of herbal products in the country. Safety assessment is very important and set guidelines to put things in order. As stated by the World Health Organization Global Report on Traditional and Complementary Medicine 2019, TNCM can make significant contribution to the goal of universal health care by being included in the provision of essential health services, improving the equitable access to safety, quality, and effective TNCM services can potentially meet communities' need and build sustainable and culturally sensitive primary health care. Herbal medicine being part of this traditional practice, coupled with healthy lifestyle and nutrition can help in the optimization of our patients' healing. After all, Thomas Edison said, "The doctor of the future will give no medication, but will interest his patients in the care of the human brain, diet, and in the cause and prevention of disease." In closing, the Philippine Medical Association and under its wing, the many doctors in the front line are one in the efforts of other stakeholders in the equitable delivery of health services and the provision of health care to our fellow Filipinos. We are for the safe and efficacious utilization of herbal medicine that underwent meticulous research and were endorsed for its proven benefits and safety. We likewise support the efforts of those in charge of the herbal medicine control and regulation in the country. We adhere for the guidelines and we respect the view of the people who value the common safety above personal interests. We hope that collaboratively we can optimize the utilization of our resources for a healthier nation. In this pandemic, we heal as one. Mabuhay po ang 3rd Philippine Herbal Medicine Summit. Mabuhay po ang Philippine Medical Association, PMA, working together as one. Maraming salamat po.
Thank you, Dr. Atienza. Our next speaker is Raquel Echage of the Board of Investments Perspective. Director Echage is a licensed chemical engineer from Pamantasang Lungsod ng Maynila with an MBA degree from the Ateneo Graduate School of Business. She is currently the OIC Director of Resource-Based Industry Service at the Philippine Board of Investments, supervising industry development and promotion of agriculture, agro-processing, fishery, forestry, energy, mining, and cement sectors.
Dr. Cecilia Maramba Laserte, Director of the Institute of Herbal Medicine, University of the Philippines in Manila, and her team, other resource speakers, researchers, medical practitioners, and other participants to these prestigious events, good evening. I am happy that the Board of Investments has been invited to this event. I will take this opportunity to introduce our agency to the medical community and to the other participants. And tonight, I will be talking about areas for growth and cooperation, Board of Investments perspectives. The Board of Investments is an attached agency of the Department of Trade and Industry, or DTI, and the lead agency for investment promotion in the Philippines and responsible for industry development. As mandated under the Omnibus Investments Code of 1987, the BOI publishes the Annual Investment Priorities Plan, or the IPP. It assists Filipino and foreign investors to venture and prosper in desirable areas of economic activities. It administers the granting of fiscal incentives to registered projects that qualified under the IPP. Among government agencies, the BOI has a unique responsibility of developing local industries and enhancing their competitiveness. Since 2012, the BOI has initiated the crafting of the industry roadmaps by relevant industry associations to help the government in addressing gaps through proper interventions. Since that time, the NHP sector, headed by the Chamber of Herbal Industries of the Philippines Incorporated, or CHIPI, collaborated with the Board of Investments to craft the Natural Health Product Industry Roadmap. This roadmap is now the blueprint of the industry and the guide of the government in identifying the necessary assistance or interventions for the sector to enhance its competitiveness. The NHP industry envisions to be a sustainable, inclusive, and globally competitive industry by providing safe, innovative, superior quality, effective, and affordable products that nurture both people and animals' health in every market served. Together with the Health and Dietary Supplement Association of the Philippines, or HADSAB, CHIPI has the following strategies. One is to consolidate and coordinate the NHP industry stakeholders, also to initiate a national research and development agenda for the Philippine NHP industry. It wanted to continuously have or conduct product development as well as capacity building for its stakeholders. The industry also wanted to identify and target possible market for the Philippine NHP industry. This is in collaboration or coordination with the DTI, particularly the BOI, and to conduct a consolidated strategic marketing program for each of the identified export markets. What has been done so far? From the time that the NHP industry roadmap has been completed and submitted to the Board of Investments by CHIPI, These are the activities initiated by both BOI and the private sector, CHIPI, and other government agencies to address the gaps in the industry. Ease of doing business is one of the important aspects of encouraging more investments in the country. The BOI, together with CHIPI, had a high-level meeting with the Food and Drug Administration. As a result of the meeting, the following were considered in the development of a simplified processing for license to operate or the LTO and certificate of product registration or the CPR. The pre-assessment stage is incorporated in the proposed FDA Citizens Charter that was submitted to ARTA. LTOs and CPRs will be available for printing by the applicants. It used to be mailed to the company. Now, it will be available for printing by the applicants. A new LTO online system and procedure has been considered for implementation last year. FDA also considered the deputization of the inspection of micro-scale low-risk food processors to the local government units or LGUs, with the Quezon City local government as the pilot implementation site. Through the BOI initiative, NHP products were proposed for inclusion in the 2019 Philippine Standard Classification Code or the PSCC. This will enable NHP products to have individual classification in the code for easy identification. These are some of the NHP products, around 9 of them, that were included in the 2019 Philippine PSCC. One is Sambong leaves powder, guava leaves powder, Chaanggubat tea, Yerba Buena tea, Nyug-Nyugan, Acapulco herbal tea, Ulasimang bato or pansit-pansitan, Moringa dried leaves, and Moringa powder. In terms of industry promotion, the BOI produces investment decks, value propositions, and company pitches for presentation to prospective investors and distribution to our commercial attaches who are posted abroad. We provide assistance to micro, small, and medium enterprises who are producers or processors of NHP through collaboration with big herbal exporting company to include products of these MSMEs for export. We closely coordinate with the DOST Philippine Council for Health Research and Development or the PCHRD to identify new herbal medicines or supplements that have potential for growth for purposes of industry promotion and updates on new products to treat patients with COVID-19. In the international front, the BOI included discussions on NHP products during the Philippine-Russia Joint Committee Trade and Economic Cooperation and the Philippines-India Working Group on Trade and Investments, particularly for curcumin. These are only two of the most recent joint or bilateral undertakings of the Philippines with foreign trade partners. BOI would also be including NHP discussions in similar future engagements through the Department of Trade and Industries, Bureau of International Trade Relations. In 2020, the BOI allotted a budget for the conduct of the farmers' training on basic farming and processing of turmeric based on the proposal from the Chamber of Herbal Industries of the Philippines or CHIPI. However, due to COVID-19, this activity has been pushed back and would be conducted once state of emergency due to COVID-19 has been lifted and trade restrictions or travel restrictions to the provinces has been removed. For 2021, the BOI is proposing the following activities. We have proposed a study on historical perspective on development of the herbal medicines in the Philippines the identification of key success factors focusing on the cases of Lagundi and Sambong herbal medicines or supplements, and simulation on how to replicate the success in terms of the case studies on two new and emerging herbal medicines or supplements. This study is being coordinated with UP Manila, I know with also with DOST, PCHRD, and of course GP. Secondly, we want more industry promotion activities, particularly of new and emerging NHP products. We will be conducting market study with DOST, PCHRD, and the formulation of company pitches for purposes of investment promotion. And of course, industry promotion through international collaboration and cooperation using venues for future engagement of the Philippines with bilateral trade partners. The 2020 Investment Priorities Plan is a list of economic activities where investments are to be encouraged and may be entitled to incentives under the Omnibus Investments Code of 1987, otherwise known as the Executive Order Number 226 as amended. The IPP is prepared and formulated through consultations with appropriate government agencies such as the NEDA, DOF, DA, DOE, DOT, DOST, DICT, and DOTR, and of course the private sector. The 2020 IPP, which is a 3-year rolling plan, was approved by the President on November 18, 2020 and took effect on December 6, 2020. It will be subject to annual review to determine or to consider other developments in the industry in the future. For NHP and herbal medicines and other related activities, the following are the activities or projects that may qualify for incentives under the 2020 Investment Priorities Plan. First is the manufacture of medicines, including the manufacture of raw materials to produce these medicines and the machinery and equipment that will be used in the manufacture of medicines. Investments, second activity, are investments in activities supportive of government programs to generate employment opportunities outside of congested urban areas. Programs of the government such as Balik Probinsya, activities that may be existing right now but wish to expand its capacity. Third is the commercial production and processing of agriculture and fishery products. Fourth is this is a related activity, which is the establishment of hospitals, medical and healthcare facilities, including quarantine and evacuation centers. Of course, most important activity for the promotion of the herbal medicine is R&D or research and development activities. Also, in clinical trials that include drug trials, we also qualify these projects for purposes of incentives under the 2020 IPP. Now, we also promote commercialization of locally undertaken R&D activities. We know that the DOST and other relevant government agencies, including private sector, has many R&D products and services that are yet to be discovered by the investors or yet to be commercialized. And therefore, the Board of Investments is encouraging investments on this through the provision of incentives under the 2020 IPP. The last but not the least are activities that are for exports. The production or the manufacture of NHP product and herbal medicines that are geared for the export market, including those that will supply the raw materials and the services of these exporters. All of these activities are included in the 2020 IPP and may qualify for incentives under EO 226. These are among the fiscal incentives and non-fiscal incentives that are under the EO 226. Two of the major incentives right now that are being administered by the BOI are first, the income tax holiday or exemption from paying the corporate tax, which is right now at 30%. If the project has been registered by the BOI as non-pioneer, meaning that there is no new technology being employed by the company, or the product itself is not new and untried in the Philippines, then the project can be entitled to the 4 years income tax holiday. For those that are pioneering or employing new technology that are not tried in the Philippines, or producing products that are not yet produced in the Philippines or tried in the Philippines, we provide a maximum of 6 years of income tax holiday. Second major fiscal incentive is the duty exemption on the imported capital equipment, including spare parts and accessories. The project or the company needs to pay the 12% VAT, but the duty would be exempted. Now, for those that are into commercial production of agricultural product, this third incentive would be applicable to them. The tax and duty exemption on importation of genetic materials. We know that breeding stocks is for livestock, but for other agri products, it's the genetic materials. And a fourth major incentive is the exemption from warfage dues and any export tax, duty imposed, and fees, and these are available for export producers. For the non-fiscal incentives, we have that unrestricted use of consigned equipment. If the project has a mother company from abroad, they can import the equipment on a consignment basis. They will not have to pay the VAT and the duty, but there is a condition that for a certain period of time, this consigned equipment will be returned to the mother company. We also we also allow employment of foreign nationals, those that are uh, in the possession of a CEO or treasurer of the company. These are being allowed for the project. The Bureau of Customs also provides simplified customs procedures for registered companies with BOI. And for export producers or export-oriented projects, they can have an access to bonded manufacturing warehouse. Okay. Should you have further questions or clarifications, the Board of Investments are welcomes you to email us or pay us a visit in the office or give us a call. My number, direct line, and email address are in the screen right now. You can just email me or call me in the direct line, and I'll be willing to uh, provide you with clarifications on whatever questions you have, or if you have a proposed project, we can sit down and discuss your project so that they may qualify for incentives under EO 226 or the 2020 IPP. Thank you very much and good evening to everyone. Thank you for listening.
Thank you, Director Echage. We will now proceed to Dr. Eleanor Roquel on the farmer's perspective. Dr. Roquel was awarded as one of the 10 outstanding entrepreneurs of 2010 by the Entrepreneur Philippines Summit Media. She advocates for community development and has created many campaigns for health service and information to where it is most needed. To date, Dr. Roquel is an executive officer of her own marketing company, a CSR consultant and custodian of corporate shared values, and a medical director of a multidisciplinary clinic with a diagnostic laboratory and a dialysis center. Together with her husband, Patrick Roquel, they manage Global Habits Corporation, GH Nutripharma, and Biopharm and Natural Health Ingredients Corporation, Binhi Corporation, an FDA-approved herbal raw material processing plant. Here is Dr. Eleanor Topel.
Good evening. We have come so far. We have blended in this life we now call new normal, and we are slowly beating the pandemic with new opportunities for growth. That is why I'd like to congratulate Dr. Cecil Maramba Lasarte of the Institute of Herbal Medicine, the National Institute of Health, UP Manila, her team, Plemex, Binhi Biopharm, New Market Link, for making the 3rd Philippine Medical Herbal Summit possible. I'd also like to congratulate everyone joining us online on digital platform. You see, your attendance forms shaping the new frontier of medicine, which is herbal medicine in the Philippines that aims to heal by first ensuring quality herbal medicine through research, regulation, and cooperation. As a stakeholder, I'll be presenting growth on cooperation on the farmer's perspective. For me, the best way to do that is to bring you to the farm. Kaya lang, hindi natin magagawa yun ngayon. That is why our company created a short video so that you can immerse with me to the farm. The farm is situated in Natatas, Batangas, near Taal Lake. Yes, where the active Taal volcano is. The farm is majestic. It is breathtaking. You'd see the impact of how research and regulation contributed to the growth of that farm. So, tara na. Let's see the video. In the video, you'll see 3 phrases that says, I plan, I share, I save communities. When you do so, please shout out and feel, internalize. I plan, I share, I save communities, because really, you can do so. Farming is beyond planting. Farming is founded on good research. A farm to be able to grow should know how to read research and apply to the farm. Planning is helped by good research and should follow regulations. That's how to do it. Why? Because the research gives you the knowledge to know and understand the features and benefit of a particular plant that you are going to use to create medicine, herbal medicine. I always believe that the effectivity of an herbal medicine is founded on the source. The source is the plant that you take care and nurture in the farm. I plant. When a farmer plants, he doesn't just plant for fruit that bears, that gives you food, or harvest leaves that gives you a medicine to heal. Actually, in my heart, a farmer plants faith. He plants seeds of hope. With the right amount of sunlight and water on a good soil, unending prayer, our good Lord ensures the right harvest. That's planting. In the farm, you see people sharing. I share. The most generous people I know are farmers. They have so little, but they give so much. They do not just share sweat and time, but they share passion to nurture and take care of plants. Without the farmers, we won't have good harvest. The farmers share their lives. They even share their families. In the video, you will see young people working in the farm. These are children of farmers, and they chose to stay with us because they know that in farming, there is dignity of labor and there is sustainability of income. That amidst Taal volcano eruption and then pandemic, we are still able to put food on the table, clothing, shelter, and give education. Because really, farming is pandemic proof. There's so much to learn in the farm. That is why our mission was founded on how we do things in the farm. Our mission is passion to sharing. That's why we are able to create our own advocacy. It's called Puno Sagip Buhay. And everything started and evolved in the farm. In the farm, we save communities. We save lives. If you are planting something and you know that this can cure and this can help, it is your responsibility to share it. So that whatever it is that you do, your purpose is met. To save lives, to save communities. Farming for me is a holistic approach of taking care of self, of taking care of your neighbor, taking care of the community, taking care of the environment, and ultimately taking care of the earth we live in. We are living in interesting times. Borders are closing. So countries needs to be producing their own food and producing their medicine. People are going back to their country where they originated. And we in the Philippines have the best doctors, the best engineers, the best planners, the best workers. We are gifted with the right soil, right amount of sunlight, right amount of water. We are gifted with many plants and faunas. All we need to do is to harness everything and create something that when the borders of the countries open, the Philippines will be able to introduce and to offer quality products from our own produce. Quality herbal medicines na ni-research, na pinag-aralan, na napatunayan at sumusunod sa tamang regulasyon. All this we can do. And you know, lahat yan we learn from the farm. Again, I plant, I share, I save communities with these hands and fingers. You too can plant, share, and save communities. So before we start the video, I hope I have awakened the farmer within. Tara na po sa aming farm.
Thank you, Dr. Roquel. Our next speaker is Dr. Annabel Pabiona de Guzman on Regulatory and Funding Institute's perspective. Dr. de Guzman is the Director General of the Philippine Institute of Traditional and Alternative Healthcare. She earned her medical degree from the De La Salle University College of Medicine, her master's in hospital administration from the University of the Philippines, and her master's in medical education from the University of Surrey Guildford in Surrey, United Kingdom. She was a diplomat at the Department of Family and Community Medicine, UPPGH, from 1987 to 1989. Prior to being the Director General of PITAC, she has been a medical center chief for various hospitals in the country. She is married to Dr. Christopher Ray G. de Guzman, an allergy and asthma specialist, and a mother to Chris John David de Guzman, a multi-awarded international violinist.
Good evening, everyone. I'm Dr. Annabel Pabiona de Guzman, Director General of the Philippine Institute of Traditional and Alternative Healthcare. I'm going to talk to you tonight about the areas for growth and cooperation from the Regulatory and Funding Institute perspective. So the Philippine Institute of Traditional and Alternative Healthcare is a government-owned and controlled corporation which is attached to the Department of Health and was created under RA number 8423 or the Traditional and Alternative Medicine Act or TAMA law of the 1997. So, PITAC's vision is people's health through traditional and alternative healthcare, and our mission is to lead in the research and development, promotion and advocacy, and development of standards on traditional and complementary medicine, and we ensure its accessibility, availability, sustainability, and integration into the national healthcare system. So in September of 2012, the Governance Commission for GOCC issued a Memorandum Circular 2012-10 which reclassified PITAC as a research institute with a primary purpose and objective of gathering information for the education of all people for the specific field of traditional and alternative healthcare. So the functions of PITAC according to RA 8423 are the following. Number 1, to encourage scientific research on and develop traditional and alternative healthcare systems that have direct impact on public health care. Number 2 is to promote and advocate the use of traditional, alternative, preventive, and curative healthcare modalities that have been proven to be safe, effective, cost-effective, and consistent with government standards on medical practice. Number 3 is to develop and coordinate skills training courses for various forms of traditional and alternative healthcare modalities. And number 4 is to formulate standards, guidelines, and codes of ethical practice appropriate for the practice of traditional and alternative healthcare, as well as in the manufacture, quality control, and marketing of different traditional and alternative healthcare materials that are natural and organic for the approval and adoption by the appropriate government agencies. Number 5 is to formulate policies for the protection of indigenous and natural health resources and technology from unwarranted exploitation for approval and adoption also by the appropriate government agencies. And number 6 is to formulate policies to strengthen the role of traditional and alternative healthcare delivery system. So all these functions of PITAC are in the 3 major final outputs of PITAC. And DBM gives us our GAA through major final output number 1, research and development. This has the biggest chunk of our budget. MFO number 2, which is technical advisory and social advocacy services. And MFO number 3, which is the regulation of traditional and alternative medicine practice. So, in order to ensure the quality and appropriate use of herbal medicine through PITACs, we have to discuss first about research and development. So in research and development, we have research on safety and efficacy of herbal medicines. We also have research on documentation of traditional knowledge in health. We also commit to ensure proposals that they observe access benefit sharing mechanisms. We also have developed or are developing the very first Philippine Herbal Pharmacopoeia, and we have also involved ourselves in technology transfer development. For social advocacy and training, we conduct BHW or LGU trainings on proper collection, harvesting, storage, use, and dosage preparation of our herbal medicines. We also have published books, and these are also the results of our research and development. We have Encyclopedia of Medicinal Plants, Volume 1, 2, and upcoming is Volume 3. We also have a book in the vernacular, Patnubay sa Paggamit ng Halamang Gamot. And for the last but not the least, we have standards and accreditation. So here we formulate standards, guidelines for the manufacture, quality control, and marketing of different traditional and alternative healthcare materials, our natural and organic products, also for the practice of our practitioners, for the approval and adoption by the appropriate government agencies. So collaborations, we usually collaborate with national government agencies, and part of our board, we have the Department of CHED, Department of Education, of course. Foremost is the Department of Health, in which our board is headed by the present DOH Secretary. We also have DNR, DA, and a lot of other private individuals who represent the different modalities, and also the academe. So we also collaborate with local government units and barangay health workers, with the academe, with private institutions, and even with non-government organizations. So what are the areas for growth and collaboration for PITAC and DOH? So we have here a WHO Global Report on Traditional and Complementary Medicine of 2019, and I would like to discuss the main difficulties faced by the member states. So these are the foremost four top difficulties. So number one, of course, is the lack of research data. Number two is the lack of financial research funding. Number three is the lack of mechanisms to monitor the safety of our TNCM practices, and the lack of education and training for our TNCM providers or our practitioners. So for our role as stakeholder, as a funding agency, what are the common difficulties? Sometimes we lack researchers to fund, and sometimes we have enough researchers, but we may lack the financial support to fund these researches. And we also lack mechanisms to monitor and regulate the outcome of these researches. So what is our role as a stakeholder in herbal medicine, and how do we address these difficulties? So these are the areas for collaboration as stated in the WHO Global Report on Traditional and Complementary Medicine 2019. So these areas for collaboration is where support is badly needed. So we need to have a common and shared knowledge for research and evaluation of traditional and complementary medicine as related to safety, quality, and efficacy. We also need to share information on regulatory issues. We also need to provide research databases. We have in PITAC the Traditional Knowledge Digital Library, but it has to be expanded and updated. So we also need seminar workshops about national capacity building on safety monitoring of herbal medicines. We also need seminar workshops about integration of TNCM and use of herbal medicines and how they could be integrated into the National Healthcare Service Delivery System. We also need seminar workshops on developing national policies on the use of herbal medicines. And we also need provision of technical support to promote safe and effective use of indigenous traditional medicine in public health care. We also need cooperation channels between national health authorities. So at present, PTAC is cooperating with the UST and FDA in the regulation of our traditionally used and herbal medicines. We also need to provide guidance and information for the public at the community level on traditionally used and herbal medicines. So this is part of our social advocacy. So it is very good to note that during the last ASEAN Cluster 3 meeting, which was hosted by Thailand, I would like to give the top priorities for 2021. And number one is traditional and complementary medicine. During the 7th ASEAN China meeting also, they also declared that they want to improve the quality of traditional and complementary medicine. So one of the sources of our funding could also be ASEAN. So how do we strive for balance? Our ultimate goal is to provide improved healthcare through provision of quality, cost-effective, accessible, and acceptable traditional and complementary medicine products and services. How do we do that? Of course, research is number one because we have to provide evidence-based information. I also believe that regulation is very necessary. And once we have our results, this has to be integrated into the national healthcare delivery system. Then we have to protect and sustain our resources, both in traditional knowledge and in the use of herbal medicines. And we also have to implement community level. We have to incorporate it into the community and use it for community empowerment. So these are what we should do. We should cooperate more and compete less. We should complete and not compete because as Henry Ford has said before, if everyone is moving forward together, even success takes care of itself. So I would like to leave with this verse in Galatians 6 verse 9 which says, "And let us not be weary in well-doing, for in due season we shall reap if we faint not." So thank you very much. This is the Philippine Institute of Traditional and Alternative Healthcare. This is our building, a 5-story building at the East Avenue Medical Center compound. And you can reach us to the following numbers and also through our email. May God bless us all. Good evening.
Thank you, Dr. De Guzman. Next, we have Director Jesusa Joyce Sirunay on the Philippine FDA's perspective. She is currently the Director of Center for Drug Regulation and Research of the Philippine Food and Drug Administration. Her government service began as pharmaceutical researcher, then as senior drug evaluator, before assuming management positions in various timelines, such as field cluster director in various parts of the Philippines, as head of the GMP's inspectorate, as head of the distribution inspectorate, and as head of the marketing authorization. Her repertoire also covers experiences in international collaborations as former OIC, FDA International Affairs Office, media relations as former FDA spokesperson, former quality manager of the FDA Quality Management System on ISO 9001, initially for 2008 version and recently for the 2015 version. On a sane harmonization in healthcare sector representing the FDA Philippines as head of delegation and APEC as delegate. Her publications include, among others, lead author in several scientific articles published in peer-reviewed international journals covering pharmaceutical science, chemometrics, such as factorial designs, central composite designs, and liquid chromatography. She is a recipient of many awards, such as the Certificate of Appreciation as Chairman of the Science and Technology Advisory Council, Belgium chapter, Certificate of Commendation for the exemplary role played as Food and Drug Regulation Officer, and Certificate of Recognition as Quality Management Representative for exemplary performance.
Good evening, ladies and gentlemen. And it is our pleasure to be here at the third Philippine Herbal Medicine Summit, ensuring quality of herbal medicines through research, regulation, and cooperation. We'd like to thank the Institute of Herbal Medicine, National Institutes of Health, University of the Philippines, Manila, for inviting us. I am Joyce Sarunay, the Director for the Center for Drug Regulation and Research at the Food and Drug Administration, Philippines. My presentation will be divided into these sets. So we have the introduction. I'll run you through Food and Drug Administration, Philippines, then the regulation for herbal medicine, TRADMED, and then the regulatory processes. Now for the introduction, the mandate of the Food and Drug Administration is to ensure safety, quality, and efficacy of health products. Now for the food, it's only safety and quality, but for the drugs, there is efficacy. Now, we issue license, we issue registration. We also conduct post-market surveillance, and now for the COVID vaccine, we issue EUA. Now, we have, for all of these authorization, it is to ensure safety, efficacy, and quality of the health products. Now, there are different products that the FDA is covering. There are food products, could be geriatrics, could be the conventional food, could be health supplements, or what we call the food supplements. We have the drug products. It could be vaccines, biologics, pharmaceutically formulated veterinary products, or health the herbal products and the traditional medicine. So we have also the cosmetics and then the devices. We have also toys, and we have household hazardous substances. As part of the Department of Health, the Food and Drug Administration ensure public safety, even in these times of the pandemic, by making sure that the products are safe, good quality, and effective. Now, for the Food and Drug Administration's organizational structure, we are headed by the Director General, Undersecretary of the Department of Health. We have Dr. Rolando Enrique Di Domingo. And then we have for the Office of the Deputy Director General for Internal Management, I'd like you to focus on the Legal Services Support Center, because this is where reports of violation will be filed, and administrative cases, criminal cases that will be filed in court, orders to close will be emanating. For revisiting of existing policies or making of new policies, we have the Policy and Planning Service. Now on the other side, for the Office of the Deputy Director General for Field Regulatory Operations, we have for the North Luzon cluster, covering regions 1, Ilocos, 2, the Cagayan Valley area. We have regions through the central, region 3, the central Luzon, Pampanga, and the rest. And we have the Cordillera Administrative Region. For South Luzon cluster, we have 4A Calabarzon, 4B Mimaropa. Then we have region 5, the Bicol region, and NCR. For the Visayas cluster, we do have regions 6, Iloilo, Bacolod, regions 7, the Cebu, and the provinces, Central Visayas, and we have the region 8, Samar and Leyte. Then for the Mindanao East cluster, we have region 10, the Cagayan de Oro areas. Region 11, we have the Davao regions, and we have region 13, Caraga. For Mindanao West cluster, we have region 9, Zamboanga Peninsula, and we have region 12, Coronadal and others. Originally, ARM was included, but now with the establishment of BARM, Mindanao West cluster has only 2 regions. And then we have the laboratory services from the different parts of the Philippines. So we have the Alabang, which is here in the central office, and then we have the Cebu laboratory, which is in Hagubia, Mandawi, and then we have the Davao laboratory, which is in Tagum, and then we have the physics laboratory, which handles for the medical device and radiation health. For the different product lines, we have specific centers handling it. So we have the Center for Food, Center for Drugs, Center for Cosmetics, and then Center for Device and Radiation Health. Now for the regulation, for the regulation of traditionally used herbal medicines, we have Administrative Order 184 Series of 2004. This is where you can read the policies, the guidelines, and registration requirements. Now, the preparation will have to have an experience, a traditional experience of long usage, which is at least 5 decades or 2 generations, noting each generation is 25 years. For products originate from other countries, there has to be an authentication of the documents coming from the Philippine consulate of where the product is coming from. Now, for herbal medicine, we have Administrative Order 172 Series of 2004, where the policies and guidelines and registration requirements are explained. You can read through, you can refer it before you prepare for the license to operate and the registration itself. Now, for the herbal medicine, it has to be finished, labeled medicinal products that contain as active ingredients, could be aerial or underground, or any other plant material or combination thereof, whether in the crude state or as plant preparations. May contain excipients as well. Now, they have to have very specific therapeutic claims and could be intended for use as diagnosis, alleviation, cure, or treatment of disease and promotion of health. But what it should not is that these preparations containing plant materials combined with chemically defined therapeutically active ingredients, including chemical defined isolated constituents of plant use in conventional or Western medicines, are not considered to be herbal medicine. Now, for the labeling requirements, the herbal medicines and traditionally used herbal medicines are covered. So they have to follow the generic labeling requirements following Administrative Order 2016-0008. Now, we do have partnership with the Philippine Institute of Traditional and Alternative Healthcare, we call PITAC, for the development of the Philippine Herbal Pharmacopoeia. Now, it's not only in the Philippines. The Association of Southeast Asian Nations have also a harmonized requirement that will be followed by the 10 member states. So it will be Philippines, Indonesia, Singapore, Malaysia, Thailand, Brunei, Vietnam, Lao PDR, Myanmar, and Cambodia. The ASEAN was established in 1967. Now, for the ASEAN Consultative Committee for Standards and Quality, ACCSQ, it was established in 1992. Then there are product working groups that are handled by the ACCSQ, and one of which is the traditional medicines and health supplements. So it's called ACCSQ TMHS Product Working Group, which was established in 2004, conceptualized in 2004, and between 2004 to between 2005 to 2014, there were a lot of discussions, harmonization, development of ASEAN common technical requirements, good manufacturing practice, capacity building, and others. Between 2015 to 2020, signing of the agreement, it was a trial run, capacity building amongst the 10 member states, and the small medium enterprise. They were how they will be assisted. It is through this 5-year span that there was a development done. And then it is expected to be fully implemented in 2021. Now, just basically alignment of regulations amongst the 10 member states. And currently, there are 9 guidelines developed. Now, as defined, any medicinal product for human use consisting of active ingredients derived from natural sources, plants, animals, or minerals in accordance with traditional medicine principles, and shall not include any syrup preparation, vaccines, any substance derived from human parts, or any isolated or characterized chemical substances. So these are the guidelines. For Annex 1, you'll have the inclusions or exclusions. Annex 2, you have the additives. Annex 3, you have the limits of contaminants. Annex 4, you have minimizing the risk of transmission of transmittable spongiforms. And then you have Annex 5 for the stability and shelf life. Annex 6, you have safety substantiation. Annex 7, you have claims and claim substantiation. Annex 8, you have GMP. And then Annex 9, you have labeling. Now, for the regulatory processes, first let us discuss licensing. For FDA, there are 2 main authorizations that we are issuing. One is the license to operate, which I'll articulate first, and then you have the certificate of product registration. But if it involves manufacturing, there's a third certificate, the certificate of good manufacturing practice compliance, that the company has to present to us, that from start to finish, they have complied with how to prepare the product the same as yesterday, today, and tomorrow in compliance with the technical specification of the product. Now, for the license to operate, it is an authorization issued by the Food and Drug Administration to a particular establishment and to a particular address of that establishment. So you cannot, in one license to operate, there is one site declared, and you cannot have multiple sites. Then you will be issued a different LTO if you have multiple sites. Now, these are the categories. So you have, as a manufacturer, could be as a trader, could be as a distributor. You import a product, you'll be an importer. You export a product, bring the product from the product of the Philippines to other countries, then you'll be an exporter. But if you want to be a wholesaler, like you get different products and then deliver it in Mindanao, so you become a wholesaler. Now, if you're going to conduct a clinical research, then the contract research organization has to have a license with us as well as the sponsor. Now, these are the requirements for the initial application. The validity is for 2 years. Now, for the fees, and then if you want to go to our website, this is the menu that you will see. Let's go to the e-service. So these are where you have to click. If you are a contract research organization, you'll click this, then there will be a drop down of how you're going to fill up. Now, these are the issuances from wherein you can base on why we require this and that, and how much is the fee. Now, let's go to registration. As I've presented earlier, we'll have the CPR at the end, but the CPR is product specific. Only the manufacturer, the trader, and the distributor can apply for a certificate of product registration, not the outlets, the CRO, or the sponsor. Now, the CPR is an authorization for the manufacture, importation, exportation, sale, offer for sale, distribution of pharmaceutical products. Now, before you can apply for a CPR, you have to have a license to operate. Now, for that license to operate, then you have to declare, if you're a licensed to operate this as a manufacturer, then the moment you finish manufacturing and finishing the pilot and the rest, then you can apply for a a CPR so that you can manufacture commercially for availability in the market. Now, since this is a marketing authorization, now this is where the products will be based. If you're going to do a market surveillance, we have to check whether that particular product is approved to be placed in the market, or if it does not have a CPR. When it does not have a CPR, then appropriate regulatory action will be done. Now, for the classifications, we have household remedies over the counter. Now, for the traditional used herbal medicine, traditional use herbal products, we have over the counter. We have also herbal medicine. Then we have also for prescription herbal medicine and traditionally used herbal products. Now for the processing time is 1 year for the initial, and the validity will come later. So the validity is 5 years, and the fees that you have to pay. Now for the requirements, the basis is Administrative Order 66 series of 1989 for both the herbal medicines and the traditionally used herbal products. Now again, the menu, you have to click drug. Then you will see it's a drop down. We'll go to downloadables. So these are the things that you will see. You have to click the drug registration requirements. You will see these requirements. Herbal medicine, traditional use herbal products. Now these are the requirements. Once you open it up, these are the requirements that you have to present. Now these are the legal basis for the issuance of the certificate of product registration. These will be all, and thank you very much for your time. I will now stop sharing.
Thank you, Director Sirunay. We will now proceed to the open forum. For any questions, please use the raise hand function, or at least type in your questions at the Q&A box or through the chat box now. For our first question from Ms. Rescober, inquiring about the use, or if herbal teas with tested antioxidant properties are considered an herbal supplement or food product. Anyone from our panel can answer? So again, the question goes, they're curious to ask if the herbal tea with tested antioxidant properties are considered an herbal supplement, or whether they're considered as food products.
Hello, good evening. For us in the Food and Drug Administration, it would depend on the product, what is the component, and what is the claim. For example, senna leaf tea is not considered as a food supplement or a food product, but is considered as a drug product under the Center for Drugs.
Okay, thank you, ma'am. Also, a related question directed to the FDA would be, will the FDA consider limited claims labeling on supplements?
I am from the Center for Drugs and Regulation and Research. We do not register or regulate food supplements, so I could give the question to the Center for Food and provide you an answer through your organizer.
Thank you. Related questions regarding certain documents would be also for the drug discovery and development manuals, if they are freely downloadable, or any of our panelists who can point to a possible link for access to these manuals, especially regarding drug discovery and development and other similar resources.
Yeah, so may I respond to that? So
Sure.
actually,
This was published by the DOSTP-CHRD, and I think this is not freely downloadable in Google. So that's why my suggestion is for Ma'am Angeli Ann Rescober and other interested attendees to get in touch with PCHRD so that to request these protocols, because I'm also not sure if there is a cost to this or if this will be freely sent out to those who request it, because this is several volumes.
All right, thank you po. Uh, To our to the rest of our participants, are there any last questions that we might be able to ask our panel? For participants, you can input your questions directly either at the Q&A box or through the chat box. While waiting for any additional questions, may we request our panelists to please, if they could turn on their cameras for a quick photo and screenshot.
Tanggalin yung share. Yan. Okay. Screenshot. Whoever is there around, open your cameras. Smile, 1, 2, 3. Ayan. One more. Thank you.
Okay, one last question po. Regarding the biofarm, a question from Mr. Alan Gran. Can we visit the biofarm and what are the schedules?
Hello, sir. Yes, that has always been a question if they can come visit the farm. Opo, pwede po. You just have to maybe ask SL kasi he's one person that is the we can be point person. If you want, just let us know and we'll give you a tour sa Binhi Bio Farm. As a matter of fact, in the past, we have already entertained Koreans, Japanese, Malaysians from different universities, and they visit the farm because they would also want to know how we do it, how they could sustain their harvest, and how to bring the farm close to the farmers. Kasi akala po kasi nila, ang farm ay nakakatakot puntahan, malayo, remote, at pagkatapos ay wala kang kasama. Alam niyo po, pag mahal kayo ng farmer, lagi po kayo may kasama, hindi po kayo mawawala. Kasi sa sobrang laki po nun, talaga pong pwede po kayong maligaw. So yes po, you are very welcome. Just let us know. And you please very well.
So yeah, thank you, ma'am. A related question po is one of our participants wants to know how many communities have been recipients of the Binhi Biofarm of medicinal plants, and how we can create more of these biofarms.
How many
communities have been recipients of the Binhi Biofarm?
Maybe how many families or
community?
Yeah.
As far as the families are concerned, napakarami ang family sa isang community, Pero to run down, we have already set up, of course, the farm nearest is not at Batangas, but we also have in Pagbilao, Quezon, in Mindoro, we have in Jaén, Nueva Ecija, Dingalan, and Peña Blanca. And right now, Patrick is in Bulacan because he is meeting with the DA, Department of Agriculture, because we are also setting one in Bulacan and I think another one in Tarlac. As far as the family numbers are concerned, hindi ko na po yan mabibilang. Ang nakikita ko lang po yung nasa plantilla ng pasweldo ng isang farm. And I suppose hindi po yon yung representative ng mga family sa community na yon.
Okay, thank you po. Now, for the interest of time, we'll have the remaining questions shall be answered by our panelists through the Q&A chat box. So we encourage our panelists to, they're able to answer the questions directly and type the answers addressed to those who have inquired or have their specific questions. If we have no further announcements from the team.
Can I just interject, Paulo? I'd like to thank our PMA president for coming over, for being here in our Zoom, because we've butt heads with the PMA in the past, but we are so happy for the support that the PMA is giving us now and the acceptance of herbal medicine right now. So Benny, thank you for being here. It's very much appreciated, the showing your support for
Welcome.
the herbal medicine community.
Waiting for your next, we will have a Zoom meeting for clusters to have the herbal medicine topic kasi interested ng mga doctors ngayon.
Yes, thank you. We'll be very much excited to participate there. And also for the BOI, first time nila in our summit. And we're also excited to hear that they have so much to offer the stakeholders here in terms of support and even for investors, for the industry, and even for the researchers. No? So we're really excited that we have you in our community, and we hope to work with you in the future.
Thank you, Dr. Maramba. It's our pleasure to be joining you in your summit. Sana in the next in your future events would also be participating kasi we have a lot of programs. Government has a lot of programs for the sectors. Isa lang to sa mga sectors that we nurture so that maging competitive talaga ang herbal medicine in the country. Thank you very much, everyone.
Thank you.
Okay, now we proceed to our top 5 Plemex Herbal Medicine Research Contest winners.
We'll just be flashing our certificates. Ito naman.
For our awarding of certificates, the citation reads, the Institute of Herbal Medicine, National Institutes of Health, University of the Philippines, Manila, presents the certificate of appreciation to our speakers, Evangeline Amor, Benito Atienza, Raquel Echage, Eleanor Raquel, Annabel de Guzman, and Jesusa Joyce Selunay in recognition of their invaluable contribution as speakers at the 3rd Philippine Herbal Medicine Summit with the theme Ensuring Quality of Herbal Medicines Through Research, Regulation, and Cooperation, held on January 25 to 27, 2021. So again, we thank our panelists for their time and expertise. Our next event would be the top 5 Herbal Medicine Research Contest winners. From all the research submissions, our esteemed panel of judges selected the top 5 research posters to be presented today. Our judges are Dr. Lynn Crisanta R. Panganiban, a family medicine specialist and clinical toxicologist. She finished her Doctor of Medicine degree at the UP College of Medicine in 1984 and underwent residency training in family medicine at the Philippine General Hospital from 1986 to 1988 and during her chief residency in 1989. She underwent training in clinical toxicology at the PGH and UPCM Department of Pharmacology. She has a diploma in healthcare evaluation and management skills from the University of Toronto in 1990 and earned credit units for the Masters in Public Health at the Midwest Center for Occupational Health and Safety, University of Minnesota School of Public Health in 1998. She undertook observership in clinical toxicology at the Ontario Poison Control Center in 1990 and is currently a full professor in pharmacology and toxicology at the UP College of Medicine, Department of Pharmacology and Toxicology, and a clinical toxicology consultant at the National Poison Management and Control Center the UP College of Medicine Philippine General Hospital. She has completed several researches, a number of which have been published in peer-reviewed journals and authored or co-authored in 16 books, notable of which is the Black Book in Toxicology entitled Algorithms of Common Poisonings. Our next judge is Dr. Agnes L. Castillo, who is recognized as one of the 25 recipients of the Philippine Men and Women of Science awarded by the DOST in 2015. She was also a DOST SEI Scholarship Awardee in 2010 to 2012 and received DOST Academic Excellence Award in July of 2013. Currently, she is a faculty member of the UST Faculty of Pharmacy and the Graduate School and a resident researcher at the UST Research Center for Natural and Applied Sciences. In December 2014, Dr. Castillo was also chosen as the recipient of the Dr. Alfredo C. Santos Award, which is given to young PhD natural product scientists by Natural Product Society of the Philippines. Her works on pharmacology of natural products and its development, like Pinospora lotion and drug delivery systems, have been published in Scopus Index, peer-reviewed national and international journals, and have won research paper and poster contests. Our next judge is Dr. Jessica Ray, an Assistant Professor 7 at the Institute of Biology, University of the Philippines, Diliman. She obtained her Doctor of Philosophy in Genetics with a minor in plant pathology at the University of the Philippines, Los Baños in 2008. She is currently in charge of Molecular and Systematics Lab at the University of the Philippines, Diliman. She has completed several researches, a number of which have been published in peer-reviewed journals. We'd also like to point out that due to time constraints, only our panel of judges may give questions to our presenters. We now proceed to our research presentation.
Good day. I am Jocelyn Alvarez, and together with my co-investigators, Mr. Alan Pascual, Ms. Corneli Basco, and Dean Bienvenido Balotro, this is our undergrad thesis from the University of the Philippines College of Pharmacy entitled Evaluation of the Wound Healing Property of Formulated Ointment Using Extracted Curcumin from the Curcuma longa-Lin family Zingiberaceae powder among diabetes-induced Sprague-Dawley rats. This is the content outline of the presentation. Diabetes is a global epidemic, and in 2016, WHO projected that it will be the 7th leading cause of death in 2030. The major increase in morbidity and mortality of diabetes could impair wound healing process and can lead to lifelong complications such as limb amputations. To treat diabetic wounds and prevent further infections, antibiotic ointment is topically applied on affected areas, which could concurrently provide high local drug levels and avoid systemic adverse effects. Curcumin or deferoyl methane is an active pharmacological agent with proven antioxidant, anti-inflammatory, and anti-infective properties. It was extracted from the rhizomes of Curcuma longa L, commonly known as turmeric. Studies to date have shown that the topical application of curcumin exhibited more pronounced wound healing effects compared to the oral administration due to the greater accessibility of the drug at the wound site. However, there is still no existing formulation of a topically applied curcumin that has been tested to treat diabetic wounds at the time of this study. This study aimed to formulate the wound healing ointment from the extracted curcumin from Curcuma longa L powder and evaluate its efficacy among diabetes-induced rats. And here are the specific objectives of the study. For the methods, our methods are subdivided into the following processes. Our sample was Curcuma longa powder. Curcumin was extracted through Soxhlet apparatus. Color, odor, and pH of the extract was evaluated. Phytochemical screening of the extract was also conducted. In the pre-formulation studies, extract was subjected to stability testing where vials were exposed to the following conditions and indicated temperatures. Compatibility of the extract with white petrolatum and beeswax were also tested. TLC analysis was conducted for each vial and chromatograms were compared with the TLC of the profile of the original extract. In the trial formulation, 3 ointment formulations, 1, 5, and 10% gram extract per gram ointment base were prepared. Fusion method was employed and small-scale batch was produced. In wound healing assay, 30 male Sprague-Dawley rats were used as animal models with the following specifications. The rats were kept separately in polypropylene plastic cages for the whole study period and acclimatized accordingly for 1 week prior to diabetes induction. The study was approved by the IACUC prior to the animal experimentation. After the 16-hour fast, the rats were weighed using a calibrated balance. Diabetes was induced by administering a single dose of alloxane monohydrate intraperitoneally. 72 hours after the injection, blood samples were withdrawn from each rat's tail vein and diabetes was confirmed by measuring fasting blood glucose levels which has exceeded 200 milligrams per deciliter. On the day of the DM confirmation, Zolitil was used to anesthetize the diabetic rats and were excised with 1 full thickness of wound excision created at the dorsum of each rat. The rats were given time to fully recover and were randomized among the respective treatment groups, positive and negative control groups. For the determination of the wound healing activity of the ointment, the following parameters were measured. The first was the percentage wound closure, where the photomicrographs of the excisional wound areas were taken on specified days and the areas of wounds were analyzed using ImageJ software and was calculated with the following formula. The period of epithelialization was monitored by monitoring the number of days required for the eschar to fall off and leaving no raw wound behind. For the percentage of scar-formed wound sites were monitored visually for scar formation, redness, hyperpigmentation, and it was determined by the following formula. Histopathological analysis was performed on the 14th day and 3 representative samples were taken from each of the 5 groups by collecting skin tissue samples, and the slides were analyzed blindsidedly by 1 pathologist and 2 researchers using the modified Ehrlich and Hunt numerical scale with the following parameters. And lastly, the data were statistically analyzed using the following tests. For the results and discussion, the curcumin extract was subjected to organoleptic evaluation and the pH of 6 was measured. Presence of alkaloids, cardiac glycosides, saponins, and carbohydrates were confirmed by phytochemical screening. And for the pre-formulation studies, the extract was subjected to various conditions of light, air, and temperature, and there were no changes in color and no mold growth in 4 weeks, which suggests that the extract is physically stable also with the excipients. Thin layer chromatographic analysis was also conducted and concluded that the extracts and extract excipient mixtures subjected to different conditions were stable and compatible. These are the trial formulations with the respective percentage concentration suitably formulated into a topical preparation with the following effects. When tested for the percent wound closure, all the treatment groups performed better than the positive and negative control groups. However, the 10% ointment was the one which exhibited the highest performance since it has both the highest mean percent wound closure and rate of wound closure. In terms of period of epithelialization, the 10% ointment showed the fastest PE among the groups, and this result was consistent with the results of the percentage wound closure and the rate of wound closure, because this indicates that a fast rate of wound closure would result in a shorter period of epithelialization. For the percentage scar form, the mean percentage scar form of the treatment group were lower than that of the positive control, which shows that the scars formed among the treatment groups were smaller than those among the positive control. Here's the gross examination of wound at day 14 from each representative group. And to better understand the parameters in the modified Ehrlich-Hunt scale, a score of 0 in the scale indicates no evidence or presence of the parameters, while the highest score of 4 plus remains confluent. Here's the photomicrographs taken under the scanner objective of normal rat skin tissue and rat skin tissues from treatment and control groups at day 14. Histopathological analysis showed that among treatment groups, 1% and 5% moderate number of inflammatory cells were observed. The 10% ointment treatment group produced comparable results with the positive control, which is mupirocin, and exhibited a mild number of inflammatory cells. It is evident that in all treatment and positive control groups, there were abundance of fibroblasts in growth and collagen formation. The final formulation, which is 10% extract ointment, passed all the parameters. It was then concluded that the extracted curcumin from Curcuma longa L is stable when subjected to various light and temperature conditions. It is compatible with white petrolatum and beeswax, and they were used as excipients. The wound healing assay showed that the three treatment groups exhibited better wound healing activity than the positive control and the negative control in terms of percent wound closure, rate of wound closure, and the period of epithelialization. Among them, it was the 10% ointment that exhibited the highest wound healing activity. It is also recommended to consider other hydrophobic ointment bases and to conduct both accelerated and real-time stability studies for 6 months to establish accurate shelf life of the preparation. The use of cell lines in doing wound healing assay is also recommended to replace the use of animals and overcome inconsistencies. Performing a skin irritation test may also be done. Thank you for listening.
Okay. Paolo, can we raise our questions now?
Yes po, you can now proceed po with the questions to our researchers.
Okay. Jocelyn, this is Dr. Panganiban. Thank you for your presentation. What is unique about your research is that you're looking at the efficacy of this particular plant as far as wound healing property among diabetes-induced prey dolly rats. So my question is, were you able to establish the duration of the hyperglycemic effect of alloxan? And in so doing, because you did your experiment for a period of 14 days, were you able to document that throughout the study, your rats persistently are hyperglycemic?
Yes, thank you for that question, Doc. In behalf of my co-investigators, I would like to acknowledge the support of our thesis advisor first, who is in the Zoom meeting today, who is Dr. Dean Balotro. And in response to that question, the FBS of the rats after the induction of diabetes was confirmed to be greater than 200 milligrams per deciliter, and after the euthanasia or on the day of the day 14, it was also confirmed that the diabetes is still present, being tested with the fasting blood glucose of levels of exceeding 200 milligrams per deciliter still.
Okay, thank you, because it's something that you did not present in your data. So I thought that you didn't get a post-experiment fasting blood sugar in the animals. Thank you,
Yes, thank
Justine.
you.
Just one comment. Maybe in the title, you just forgot to write down after sprayed dolly rats. Okay,
Yes,
the
thank you.
use. Okay, thank you.
Thank you.
Good,
From our panel, are there any additional questions?
good evening, Sir Paulo. Can I address the
Yes,
next
po.
question?
Please proceed.
Hi, Jocelyn.
Hello.
Congratulations. This is Dr. Castillo. So I would like to ask your group, aside from TLC, which you have used to assess stability of curcumin and its compatibility with the ointment base, what are other methods that you may utilize to assess compatibility during your pre-formulation studies? Hi,
Thank
just
you for that question. Hello.
clear? Did you hear me?
Yes.
Okay.
Yes po. The TLC, the thin layer chromatographic analysis was used to measure the stability and stability of the extract from the accelerated TLC was used to check the stability of the of the was used to check the stability of the extract with the excipients, which are beeswax and white petrolatum. And QC was done after the stability test to confirm the QC test, or the quality control test, were used to were done to for the stability, and it passed all the parameters, which are viscosity, which I may mention is appearance, color, odor, viscosity, spreadability, spatula feel, and the stability of the extract at temperatures 28 degrees and or room temperature, and the oven temperature, which is 40 degrees Celsius.
Okay. In the pre-formulation studies, aside from TLC, we also perform FTIR and DSC or DTA. Can I also ask another question? May
Yes.
I ask? Okay. You have utilized the wound excision model. What
Yes.
are the advantages of wound excision model over the wound incision model?
Mm. Thank you for that question, because I think that the wound excision model will be more relevant in measuring the wound size of the rats, especially following our parameters, which are percent scar formed, the percent wound closure. The wound excision is more what do you call this? Is more relevant or more up for the parameters we have tested.
Okay, thank you.
Thank you po.
Okay, thank you for our panel and to our investigators. Yes, we have one last question.
Just follow up, actually related sa question ni Ma'am Agnes. This is Jessica Ray. Jessel, yung decision nyo to use the ointment, was this direct from the start you had decided already, or meron kayong pre-evaluation ng like solution, or like in the form of spray, or iba pang yung emulsion na preparation for the final product?
Yes po. As said in the presentation po, the studies demonstrated po that the topical application of curcumin has more pronounced wound healing compared to oral administration, because of owing to this its greater accessibility at the wound site. And yung curcumin po ay limited water solubility and has extensive first pass metabolism. So in that first pass metabolism, the drug will not get metabolized at the specific location in the body, resulting to reduced concentration. So we resorted to the topical application.
Yeah, I know topical, pero bakit hindi niya kinonsider niyo ba yung alcohol-based spray or something?
We haven't considered for that preparations, but it will be good for a recommendation to be included for that part po.
Okay. Yes. Yun lang, Paolo. That's all.
All right. Okay. Thank you. Thank you to our panel, and so we will now proceed to the next presenter. Thank you.
Good evening, everyone. I am Ryan C. Vilintao, and I am here to present our project entitled Lyophilized Ethanolic Extract of Rhizophora mucronata Leaves Screened for Protective Activities Against Behavioral Decline in C. elegans. Neurological disorders are diseases of the brain, spinal cord, and nerves caused by structural, biochemical, or electrical abnormalities such as infections, trauma, and neurocognitive diseases. As organisms age, neurologic systems degenerate, resulting to diseases such as dementia and Parkinsonism. Based on the 2003 National Nutrition Health Survey, there is increasing prevalence of dementia and Parkinsonism, particularly as age increases. Given the need to address these growing concerns and that approximately 80% of the world's population utilizes traditional medicine for primary care, recent studies have been exploring the use of traditional medicine. The Philippines in particular is rich in diverse flora which have potential for medicinal use. Among these is Rhizophora mucronata. Our mucronata or bakawan babae, it has a widespread distribution found in estuarine environments. It is traditionally used for diabetes, fever, and diarrhea, and preliminary studies have shown antioxidant and anticholinesterase activities. However, only studies have been conducted in vivo, none of which were done on local samples. In our study, we use C. elegans as a model organism for neurobiology. C. elegans is a free-living non-parasitic nematode approximately 1 millimeter in length at adulthood. Due to its short lifespan of 2 to 3 weeks and high fecundity, it is easy to cultivate in laboratory setting. It is an established model of eukaryotic cell function and interactions, including neurophysiology, which is of interest in our study. In particular, this graph illustrates the age-related decline in function of different C. elegans behaviors, wherein more complex behaviors decline earlier. This is the expected natural progression of the relevant neural parameters in our study. Now we ask the question, does the extract of our mucronata leaves have protective activities against behavioral decline in C. elegans? In this study, we aim to screen for potential protective activities of the lyophilized ethanolic extract, hereby referred to as LEX, of our mucronata leaves on 3 behavioral parameters in comparison to positive and negative controls: locomotion, stigmotaxis or response to touch, and nociception. Our methodology is divided into 3 phases. For the first phase, we collected 15 kilograms of mature undamaged leaves and soil sample from Pagbilao Mangrove Experimental Forest in Quezon Province and were authenticated. We proceeded with the ethanolic extraction of the collected leaves using this procedure. After garbling and air drying, we proceeded with the extraction using absolute ethanol followed by solvent removal by rotary evaporator and lyophilization. Finally, the pharmacodynamic screening was done, yielding these results. Values less than or equal to 25% of the extract were evaluated, and maximum tolerated concentration for chronic exposure was found to be 2.5% weight per volume of the extract, with an LC50 of 2.89%. Then, by means of observing these physiologic responses and behaviors shown in the screen, at low LEX concentrations, the no observable adverse effect level or NOAL was established at 2.5% Lex. In summary, these are the pharmacodynamic parameters. The low concentration Lex setup was set at 1.25%, mid concentration at 2.5%, and high concentration at 4.99%. For the behavioral assays, we have 6 treatment groups, 1 negative control, 2 positive controls, and 3 levels of our experimental groups. All treatment groups were observed on interval of 2 days for a total of 6 days. On each observation point, these response parameters, locomotion, touch, and nociception, were evaluated as described in the next few slides. The 2 positive controls used were levodopa-carbidopa, which are both indicated in Parkinson's disease, and donepezil, which is indicated in Alzheimer's disease. The negative control is the M9 buffer. Worms were individually manipulated and observed under a stereo microscope. The anterior end is operculated and rounded, seen on the left picture pointed in the arrow, whereas the posterior end or the tail is sharp and thin, shown in the right image pointed by the arrow. Using an improved eyelash stimulus, actual gentle touch sensitivity assay of the anterior is exhibited on the left, then of the posterior on the right. This is the video of the harsh touch or nociception assay using a number 00 gauge insect pen. For the results and discussion, first we look at the effect of the leaf extract on the locomotion of C. elegans. As shown, there is a general decrease in motility from day 0 to day 6 across all treatments. However, there is lower decrease in all Lex groups compared to the negative control, with comparable response to both positive controls. Based on the anterior gentle touch sensitivity assay, only the high concentration Lex group was able to attenuate the decline in anterior thigmotaxis, which is comparable to the positive controls starting at day 4. For the posterior gentle touch sensitivity assay, both mid and high concentration Lex groups were able to attenuate decline in posterior thigmotaxis, being comparable to positive controls starting at day 4. Comparing both gentle touch assays, there is greater general decline in the mean score response of the posterior assay. This may be attributed to the greater neuronal concentration at the anterior end. Lastly, for the harsh touch sensitivity assay, the scores of all concentrations of the leaf extract and the positive control were not statistically significant when compared to that of the negative control. This may be due to the fact that locomotion deficit is an earlier effect of aging in C. elegans and that sensory abilities decline later. C. elegans exhibits robust, harsh touch responses which may allow it to effectively avoid hazardous cues, aiding in its survival. Therefore, the loss of function of the nociception might have not been reached yet at the end of the experiment, explaining the persistence of the scores of the Lex groups, positive and negative control groups at days 4 up to 6. In conclusion, based on our study, we can conclude that the Lex or the extract may protect against aging-associated motor function and somatosensory behavioral decline in C. elegans. There may also be a direct exposure-response relationship with increasing concentrations of the extract. We recommend, therefore, that the potential neuroprotective properties of Rhizophora mucronata leaves be re-evaluated in a suitable mammalian model, then to isolate bioactive compounds from the extract, and finally to elucidate the exact biochemical pathways involved. This ends my presentation. Thank you.
We now proceed to the question and answer. our panel can go forward with any questions for our researchers.
Good evening, Paolo. Who is,
Ready po.
hi, who is the presenter? Can I know his name? I forgot his.
Hello po,
Ryan
Ryan
Lintao.
po.
Ryan. Hi, Ryan. Okay.
Hello po, ma'am.
Congratulations. My question is, your team determined the neuroprotective activity by observing the responses of the C. elegans on locomotion, touch, and sensation, which are all qualitative data, right? You just grade them, right? How the team removed the bias during the grading of the responses?
Hello po, ma'am. So for the uh for the uh for the assessment of the scoring system,
Yes.
initially we planned on doing inter-rater reliability test. But unfortunately, the the initial part of our project, we were not able to conduct it very smoothly because different raters uh, can ano parang there's a problem in handling the animal. So parang napapatay po nila yung animal. I think that's the problem. So we resorted to just one rater. However, we blinded the rater into what treatment group the C. elegans culture belong to.
Okay, another question, follow up. So, what quantitative measurements or methods you can suggest to assess the neuroprotection to improve your study?
Ma'am, I think for the locomotion, ma'am, I think that's a quantitative measure because we measure in terms of number of bends per minute. So for the locomotion part, we were able to at least standardize it. But for the response to touch and response to nociception, I think to be able this is how it's done in other countries. They measure the force applied, and they quantify the action of the C. elegans using neural engine computer learning technology. So that we don't have in the Philippines, unfortunately. So that's one way to be able to standardize the protocol better. But many studies have been published conducting this study in terms of scoring. So I think that's why we resorted to doing this because many authors have actually done the methodology and they were able to get reliable results naman po.
Okay, thank you.
Thank you po, ma'am.
Can I ask a question, Paula?
Please, yes po. Go ahead po.
Ryan, hi.
Hi po, ma'am. Good evening po.
Good evening. Just two questions. The first one is, what are evidences in literature that indeed C. elegans is a good screening test when your disease outcome that you want to, at the end of the day, to have benefit for this rhizophora is Alzheimer's disease. So is C. elegans the screening method for this outcome that you want to study? The second one, did you postulate on what in the rhizophora would have this effect on locomotion, on motility and touch, because you saw significant findings? So did you postulate what of those active principles would have produced such effects?
Thank you, ma'am, for the question. So for the first question po, regarding C. elegans as a screening tool,
For
actually,
Alzheimer's.
In 2016, Shu et al. Were able to propose C. elegans as a good screening tool because the problem with studying Alzheimer's disease and actually any neurodegenerative disease is that it takes too long in the life of the animal. So you have to follow up the animal up to the end of its life in order to be able to make computations regarding its uh, regarding the effect on the neurodegeneration and possible neuroprotective activity. The benefit of doing C. elegans as a screening tool is that they were able to find out that early on, the changes, the behavioral decline can be attributed mostly to its neurologic uh, function. Because at the end, beyond day 6, the decline can be attributed as well to muscular degeneration. So by screening early on in its life with the neural component being predominant in that decline, I think that makes C. elegans one of the best screening tools that we have that can also be used parang in a more shorter span of time as compared to mammalian models such as rats and mice. For the second question po, ma'am, I'm sorry, ma'am,
What this
ano po,
think would be which of those that you found as far as the active principles would have produced this significant effect, the positive effect on
we were,
yeah.
sorry, ma'am, sorry for
Yeah.
interrupting.
Go ahead.
We were able to search the literature and we were able to find some alkaloids present in the extract that can explain the effect because the according to a study by Ganthi et al. In 2014, if I'm not mistaken, or 2017, they were able to um, show anticholinesterase and antioxidant activity in vitro. So that's we're providing an possible in vivo application of what they were able to find in vitro in that study po, ma'am. So that can be attributed to the activity can be attributed to some alkaloids present in the extract po, ma'am.
Anticholinesterases. Okay,
Yes
thank you,
po, ma'am.
Ryan. Thank you.
Okay,
Thank you po, ma'am.
for Dr. Ray, do you have any questions po for our presenter?
Wala akong question, just a recommendation on if you are going to push further itong study na to, or you can put in the recommendation. I think it would be worthwhile to do a chemical assay kung ano yung mga active ingredients based doon sa mga literatures that affect the neural responses. So, baka yun yung next step nyo na for this study.
Yes, ma'am. Hopefully, we will be able to apply it because in C. elegans po, ma'am, we can have GFP or fluorescent C. elegans, wherein we can monitor certain neurons or neuronal activity. So, mas makita po namin which of the neurons or neuronal pathways involved in such behavior, which can be of potential use to for potential translatability po of the study. Thank you po, ma'am, for the suggestion po, ma'am.
Welcome.
All right, thank you to our panel and to our presenter. We now proceed to the next study.
Good evening, everyone. I'm Kitspoldi Markham, and I'm here to present our research entitled Cytotoxic and Anti-Proliferative Activity of Fractions from Policious nodosa leaf extracts against breast cancer cell lines MCF7 and MDA-MB231. Breast cancer is the most prevalent type of cancer both in the Philippines and worldwide, accounting for over 15% of total cancer cases. It has 4 molecular subtypes that vary in treatment and prognosis, and these are the luminal A, luminal B, HER2-enriched, and triple-negative subtypes. Among these, luminal A is the most common, while triple negative has the worst prognosis. The most commonly used cell lines to model the luminal A and triple negative breast cancers are MCF7 and then MDA-MB231 respectively, which is why we use these two cell lines in our study. One of the challenges with breast cancer is the inadequacy of current treatment modalities. Some of the limitations of commercial anti-cancer drugs include high cost, adverse effects, and development of resistance. There are various research areas for cancer treatment, but being researchers in the Philippines, we decided to take advantage of our country's rich plant biodiversity to try and address this problem. Probably like the other researchers in this conference, we tapped onto the potential of native Philippine plants as sources of potentially cheaper, safer, and more effective alternatives. And among the various Philippine plants, we focus on Policius nodosa, locally known as malapapaya. This plant is a native softwood tree that is found throughout the Philippines and has reported traditional uses for purpuric fever and as a contraceptive. Unfortunately, it has no published studies on its bioactivity yet. Although related plants of the same genus showed antibacterial, anti-inflammatory, and particularly anti-cancer properties, which suggests that this plant may have some bioactivity as well. Therefore, our study aimed to determine whether the components of Policius nodosa leaves can inhibit MCF7 and MDA-MB231 breast cancer cells in vitro. To do this, we first had to partially purify the leaf ethanolic extract. Then, we aimed to screen the fractions for potential bioactivity. Then we aimed to evaluate the cytotoxicity of the fractions on the chosen breast cancer cell lines. And finally, we aimed to determine what kinds of phytochemicals were present in the fractions. So here's the outline of our methodology. First, the crude ethanolic extract was subjected to silica gel column chromatography with a step gradient solvent system consisting of hexane, ethyl acetate, methanol, and water. Then the resulting fractions were screened using the brine shrimp lethality assay, and the most active fraction was further partially purified using silica gel column chromatography, this time with a step gradient solvent system consisting of chloroform and methanol. Then the subfractions were subjected to MTT assay to determine their cytotoxicity to the chosen breast cancer cell lines. And finally, we subjected the fractions to phytochemical analysis. We used the brine shrimp lethality assay as an initial screen for the fractions, since according to literature, this assay correlates well with cytotoxicity to various cancer cell lines. So the fractions that would exhibit cytotoxicity to the brine shrimp would show potential cytotoxicity to the cancer cell lines. And in this assay, fraction 7 and fraction 8 showed the highest cytotoxic activity to the brine shrimp and were the candidates for further partial purification. However, we decided to pursue fraction 8 because 1, their activities did not significantly differ at all incubation points, time points tested, and 2, fraction 8 had a 10-fold greater yield than fraction 7. So upon partial purification, we found that the subfraction, fraction 8C, had the highest activity against the MCF7 cancer cell lines. Notably, this fraction had an activity that is comparable to 2 commercial anti-cancer drugs, tamoxifen and doxorubicin, at both 50 and 10 ppm. We can also see that in contrast to the subfractions, the crude extract did not have any activity at 10 ppm, and this suggests that the bioactive compound from Policius nodosa leaf extract was indeed partially purified. Interestingly, we can see a similar trend in inhibition of the triple negative cell line MDA-MB231. Likewise, fraction 8C was also the most active fraction, and its activity was also comparable to the 2 controls at both concentrations tested. And so we have fraction 8C, the most active fraction, that is active against both MCF7, which is a luminal A cell line, and MDAMB231, which is a triple negative breast cancer cell line. And this may be advantageous for an anti-cancer drug to be effective against multiple breast cancer subtypes. So what possible compounds could this fraction contain? According to our phytochemical screening, this fraction tested positive for alkaloids, indoles, sugars, and triterpenoids. Although this qualitative test was not able to identify the specific compounds that were present in the fraction, it gave us an idea on what class of phytochemicals the bioactive compound may belong to. And we recently got the results of the mass spectrometry analysis of this fraction. And although the analysis is still incomplete, so far the identified compounds are in agreement with the results of the phytochemical screening. So those compounds so far belong to these phytochemical classes. In conclusion, we found that Polycia sedosa leaves contain bioactive compounds that can inhibit both MCF7 and MDA-MB231 breast cancer cell lines. And these bioactive compounds were partially purified into the most active fraction, fraction 8C. And these compounds exhibited cytotoxicity to the brine shrimp, and more importantly, they were cytotoxic to the breast cancer cell lines with activity comparable to two commercial anti-cancer drugs. And this fraction also contained alkaloids, indoles, sugars, and triterpenes. For further research, we recommend assessing the cytotoxicity of the active fractions against the non-cancer cell lines. This is actually the biggest limitation of our study because we can't say for certain that the active fractions are anti-cancer if we don't know yet if they are safe to non-cancer cell lines. Because of course, to prevent adverse effects, we want the anti-cancer agent to be selectively toxic to the cancer cells only. We also recommend determining the mode of cell death induced by the active fractions, and we can do this perhaps by using flow cytometry because, of course, we want the anti-cancer agent to induce a more selective type of cell death, such as apoptosis rather than non-selective necrosis. And if the results of these are promising, perhaps we can further purify the most active fraction and eventually isolate the bioactive compound. And in the distant future, if we may be able to isolate the bioactive compound, then perhaps we can elucidate the structure and mechanism of action. Before I end my presentation, I would like to acknowledge the members of the Natural Products and Peptidomics Laboratory, without whom this study would not be possible. So particularly Camille, Ate Zared, Kuya Dennis, Dr. Kiming, and Dr. Nicolas. Here are my references for this presentation. Thank you for listening.
We now proceed to the question and answer.
Okay, Kits, this is Dr. Panganiban. Yeah, thank you for your presentation. This is just some safety concerns that I have. You were using quite a number of chemicals that have been known to be for their toxic effects, like hexane, methanol, and chloroform. I just would want to know what precautions did you take when you were handling these different chemicals. And the second is how did you ensure that in the residue that there are no residues of these chemicals in the extracts or in the test material that you used for this particular study?
Yes, ma'am. Thank you, ma'am. For the first question, all the experiments that used those chemicals such as hexane, ethyl acetate, and chloroform were performed under the hood so that I would not be exposed to the gases. And to the in regards with regards to the second question, so we thoroughly dried the samples. We thoroughly dried the samples and subjected them to rotary evaporation before using them. And then afterwards, they were suspended uh, in solution, so in aqueous solution. And these chemicals, those chemicals, hexane and chloroform, are nonpolar. So we would observe a separation of layer if there were still remnants of them, and those were not observed.
Okay. Uh, why you did an ethanolic extract? Why not
Yes,
an infused extract, which is of course a much safer formulation?
because ethanol has both a polar and nonpolar group, so it would catch more of the phytochemicals present in the leaves. So since we are aiming for drug discovery, we would want more diverse uh, more diverse profile of phytochemicals to be tested, so we have a greater chance of finding an active component.
Okay, thank you.
Thank you, ma'am.
Can I ask the question?
Please, yes,
Okay.
please proceed.
Hi Marco. Good
Hi,
evening.
ma'am.
So, why your team did not include a normal cell line to check the safety or toxicity of the fraction? And follow up question, what specific assays the team has conducted to determine the anti-cancer pathways being targeted by the fraction, and also to confirm the mechanism of action of the F8, I think F8C.
Yes po. So with regards to the first question po, we didn't use a normal cell line because it was a limitation in the procurement of materials. So at the time, we didn't have a normal cell line yet. So we tried to outsource the that part of the experiment, but the results did not come in yet. And so that is a recommendation for further studies, because I think for this kind of study that is really needed. So I agree with that. And with regards to the second question, we didn't perform much experiments to determine the mechanism of action. We did do the yeast estrogen screen assay. So this assay uses a recombinant Saccharomyces cerevisiae that expresses a human estrogen receptor alpha. So it is a gene reporter assay that determines estrogenic activity, but I didn't include it anymore in the presentation because the conclusions were limited from that. So we found that the most active extract got a negative result from that assay, which only tells us that it either did not bind to the estrogen receptor, or it binds to the estrogen receptor but has anti-estrogenic activity. So we only confirm basically that it has no estrogenic activity, so it cannot have a mechanism like, for example, tamoxifen, which is a partial ER agonist. So we only ruled out that mechanism, but that conclusion is very limited. So we suggest further experiments to determine its mechanism of action.
Thank you.
Kids,
Yes, ma'am.
bakit malapapaya?
Um, Actually, my thesis. This is my undergrad thesis, and it's actually part of a bigger project by our laboratory, the Natural Products of Peptidomics, in collaboration with DOST, and they did a screening of many Philippine plants, and Palapapaya was one of those. This is why, um, even though it has limited studies, it is chosen as a candidate. It was due to a preliminary screening po.
But meron bang promising na information na nagsasabi na pwedeng, because malapapaya is a wood, right? It's a
Softwood
it's a
tree
native,
po.
yeah, it's a tree. So meron bang, is it like ethnobot data
Um,
that you based it on?
Not
This
really, ma'am.
research is okay.
It doesn't have ethnobotanical used related to cancer, but
Mm-hmm.
related plants have some anti-cancer properties, reported anti-cancer properties. But the preliminary screening, I think, used a wide range of plants, especially understudied plants, because there is some gains uh, to investigating the understudied plants. They're uh, like they have some potential. Yeah.
Okay. Sige, I will just read that the OST site for the ongoing project. So this is an ongoing project.
Yes. Po.
Thank you. Thank you, kids.
Okay, thank you to our presenter and to our panel. We now proceed to the next study.
The fight against the Big C is far from over. The global burden of cancer is estimated to have risen to 18 million new cases and 10 million deaths in 2018, with half of the new cases and cancer-related deaths coming from Asia. The most common cancer site remains to be the lung, followed by breast and colorectum. These numbers are expected to rise over the years due to various socioeconomic factors associated with industrialization and development such as smoking and unhealthy diet. Despite available chemotherapeutic modalities, treatments remain inaccessible and expensive. Herbal medicine remains to be the most accessible form of healthcare, with more than 80% of the world's population using traditional medicine in healthcare. And recently, there has been growing interest in medicinal plant extracts for anti-cancer activities. The Philippines is rich in diverse flora which have potential for medicinal use, and for this research, Alpinia elegans will be tested for its anti-cancer activity. Alpinia elegans or tagbak belongs to family Zingiberaceae and is endemic in the Philippines, where it has been used as medicinal plant for wound infections, hemoptysis, and hives. Closely related species of Alpinia elegans have been shown to have anti-cancer activity. However, there are no studies done on Alpinia elegans. Given the anti-cancer activity of its closely related species, now we pose the question, does Alpinia elegans have anti-cancer activity as well? Good evening. I am Ryan Christian Vilintao, and I will be presenting my research project entitled Morphological and Functional Effects of Leaf Ethanolic Extract of Alpinia elegans or tagbak on human lung epithelial, colorectal, and hepatocellular cancer cells in vitro. This is the objective of my study. Specifically, I would like to see the effect of the extract on cell proliferation, cytotoxicity, cell migration, and gross morphology. The methodology is divided into three phases. In the first phase, fresh leaves were collected from Infanta, Quezon. Leaf samples were authenticated and air-dried for 2 weeks for the second phase. In the second phase, dried leaves were pulverized and soaked in 95% ethanol for 48 hours. The mixture was filtered and the filtrate was concentrated using rotary evaporator. Phytochemical analysis on the sample revealed traces of alkaloids, moderate amounts of sterols, and abundant triterpenes, flavonoids, saponins, glycosides, and tannins. In the third phase, the crude extract was screened for anti-cancer activity. These were the assays done to answer our specific objectives following a post-test only control group design. One-way analysis of variance test was performed with level of significance set at 0.05, followed by two keys multiple comparison test. Differences were considered statistically significant at p less than 0.05. Anti-proliferative activity is observed using MTS cell proliferation assay in which live cells convert the substrate into formazan, thus appearing violet. Correlating with absorption, the more violet the mixture, the higher the number of cells. The extract exhibits antiproliferative activity at high concentrations compared to doxorubicin, which is the positive control, which can be explained by the crudeness of the extract. The observed activity follows a sigmoidal curve, which suggests a dose-response relationship. The extract has also been shown to be more effective based on IC50 on hepatocellular cancer cells, followed by lung and colorectal cancer cells. MTS assay, however, does not tell us if the antiproliferative activity was due to cytotoxic or cytostatic effects of the extract. To measure cytotoxic activity, LDH release assay was performed in which LDH release from damaged cells convert the substrate into purple formazan, and the absorbance was correlated with cell death. At 500 parts per million of the extract, the percent cytotoxicity is significantly different with a negative control DMSO but not with a positive control 5-fluorouracil. The same significant activity can be seen in colorectal cancer cells as well as hepatocellular cancer cells. This suggests that the antiproliferative activity of the extract can be attributed mostly to its cytotoxic effect on cancer cells comparable to 5-fluorouracil at high concentrations. In screening for metastatic potential, a scratch wound assay was done in which a monolayer of cancer cells is literally scratched off the surface of your cell culture using a pipette tip and is visualized in time-lapse. This is just to show you the time-lapse of the scratch wound assay. Cells move toward the empty space to close the gap, and the percent gap closure serves as indicator of your cell migration. After 16 hours, the percent gap closure in lung cancer cells is significantly different with both the negative control, which is DMSO, and doxorubicin at the concentration 500 parts per million of the extract. The same was observed in colorectal as well as hepatocellular cancer cells. This suggests that at high concentrations, the extract also significantly inhibits cell migration. Now, to visualize the actin filaments and nuclei, the cells were stained with phalloidin and Hoechst respectively. Fluorescence staining showed cell shrinkage to 1/16 of its original size and pycnosis to half the original nuclear length, all of which are indicative of your cell death. Cellular debris are also seen, which suggest necrosis. The same characteristics can also be seen in colorectal cancer cells as well as hepatocellular cancer cells. While the observed necrotic effects may be non-specific to cancer cells, the apoptotic hallmark suggests a modulation of cell death, which is desirable in cells with uncontrolled cellular proliferation such as cancer. In a nutshell, this study shows that the extract significantly inhibits cell proliferation and migration at high concentrations. The extract also shows hallmarks of both apoptosis and necrosis as mechanism for the extract's cytotoxic activity at high concentration. Now, to ensure that the cytotoxic activity is specific to cancer cells, it is recommended to isolate the bioactive compounds in the extract, to test the extract on normal cell counterparts, and to explore activity on other hallmarks of cancer such as angiogenesis. Going back, where are we at the fight against the big C? With growing studies on plant extracts for anti-cancer activity, the fight may be a cup of tea away. Thank you and good evening.
Okay, thank you to our presenter. We now proceed to the question and answer. Our panel can, anyone can proceed with their questions.
Okay. Paulo, can I ask a question? This is
Yes,
Dr. Pakuti. Yeah.
yes, Doc, please go ahead.
Ryan, hi again. I'm just very much interested. How did you arrive at the doses for your study? You have 3 dose levels, 125, 250, and 500 parts per million. Where did you derive all those doses? Were there studies done before in terms of like the toxicity of this particular plant, establishing MTD, and choosing your dose levels?
Hello po ma'am. So for this study po, since we were what happened po kasi is the selection of the concentration po ma'am was done by uh by doing various concentrations po starting at 10,000 ppm downwards po ma'am. So it was a standardized selection of concentrations. And so with regards to dosage naman po ma'am, we were not able to correlate it talaga because um the the terms that we've used are actually concentration,
The
which
LC50, Ryan. Ryan, the
yeah ma'am.
LC50. What's the LC50 of this plant? LC50.
For the LC50 po ma'am. Ay po ma'am, we check we check po for the we systematically check po for the different concentrations po until we were able to come up with a sigmoidal curve. Then we computed for the response with regards to the cells po ma'am. So from that po, we were able to derive a dose response curve po that appears sigmoidal. So namili lang po talaga kami ng different concentrations na standards po.
So, but you didn't do anything like establishing LD50 or the shrimp brine lethality test to just establish that you're not reaching some level of toxicity as far as the plant itself?
We were not
like ppm. That's where you're getting the significant findings. So at 500
sorry po ma'am. Sorry po ma'am.
in terms of safety kasi of the plant itself.
Yes po ma'am. Thank you po for that question. I think that's one of the recommendations of our study po ma'am, that we checked it on a counterpart cells po, so that we can be able to compare the LC50 in cancer cells in the LC50 with your normal cells po, so that we can come up po with the therapeutic index of this certain plant as compared to the cells in this study po. So that's one of the recommendations po for this study po.
Okay, thank you.
Thank you po ma'am.
Hi, Ryan.
Hello po ma'am. Good evening po.
I'm also working with Alpinia elegans, but with different activity. Based on your RRL, can you identify some secondary metabolites present in Alpinia which can contribute to your findings, the different activities like anti-proliferative, cytotoxic, and through what possible mechanisms?
Thank you ma'am for that question. Based on our RRL po, kasi there is no, as far as we're concerned, there is no parang component analysis of the Alpinia elegans as of the conduct of the study. the closest thing that we've got is the studies by Dr. Susan Kalumpang from UPLB po. And those are in application setting po. But with regards to with Alpina elegans, I think the closest source that we can get is through its closely related species, Alpinia galinga and Alpinia mutica. In Alpinia mutica, they were able to isolate at triterpene po that they found to be associated with increased responsiveness of the cancer cells to cytotoxic activity. As well in Alpinia mutica po, ma'am, I think it's a flavonoid that they were able to isolate in Alpinia mutica. So, yun po ma'am, yun po yung so far that they were able to isolate. But so far po ma'am, there are no studies on how those isolates target the molecular pathways of cancer. So I think that's another future direction of the study, to be able to find out what are the molecular pathways or molecular targets of these compounds with relationship to different cancer mechanisms po, ma'am. So,
Yes.
yun po, ma'am.
Yes, correct. What you have mentioned, these metabolites are known, proven to have antioxidant properties, and they are linked to reduction of MMPs for chondro protection activity. Thank you, Ryan.
Thank you po, ma'am.
Okay, for our panel, Dr. Faye, do you have any questions?
Paolo, wala akong question.
Okay.
Thank you.
All right, thank you. So thank you to our panel and to our presenter. Now proceed to our next study.
Good evening, everyone. I will be presenting my research, the assessment of the anti-classigenicity and nephroprotective activities of Peperomia pellucida lean HPK decoction on cisplatin-induced classigenicity and kidney injury in male albino mice. As we all know, cell replication occurs in a highly organized and tightly regulated process. However, it is affected by various factors which induces DNA damage. If a cell has an error in its DNA and cannot be repaired, it may undergo to the natural programmed cell death called the apoptosis. If not, mutations in the cell can make the cell replication proceed without order and control, thereby accumulating the genetic defect that can lead to carcinogenesis. In cancer treatment, combination of these techniques are used to remove the tumor in the body. In the case of radiotherapy and chemotherapy, the exposure of the patient to radiation and chemotherapeutic drugs often lead to other complications. If not addressed properly, it may lead to death. Cisplatin is a platinum-based anti-cancer drug, and it is one of the most widely used chemotherapeutic agents. It works by inducing 1,2 intrastrand cross-linking in the cancer cell's DNA, thereby activating apoptosis. The platinum compound is then excreted, which can affect neighboring cells. However, this process lacks specificity and selectivity because it can also affect healthy normal cells. Additionally, recent studies have shown that this anti-cancer drug itself is a carcinogen. Thus, the use of cisplatin is clinically regulated. Just like any other drug, cisplatin is eliminated by our body via renal excretion. Hence, cisplatin is accumulated in the kidney where it can damage tubular cell, causing nephrotoxicity. Among the side effects of cisplatin, its nephrotoxicity is the main limiter. Cisplatin therapy accounts up to 60% of hospital-acquired acute kidney injury, and depending on the number of episodes, severity, and duration of the acute kidney injury, it may lead to chronic kidney disease, which is a progressive and irreversible disease with no current treatment. This table summarizes the protective measures used during cisplatin chemotherapy, and as we can see, one of the options is natural products. So this indicates that scientists continuously tap the potential of medicinal plant as a primary treatment for various indications through its development as drug due to its potential high activity and relatively low cost. So Peperomia pellucida, ulasimang bato, or pansit-pansitan in Tagalog, or sinaw-sinaw in Bisaya, is a fast-growing plant that is characterized by its succulent stem and shiny heart-shaped leaf. This plant is known for its diuretic, analgesic, and anti-inflammatory properties. In the Philippines, Ulasimang Bato is one of the 10 plants endorsed by the Department of Health under its traditional health program. So these are the major constituents of this plant. Here we summarize the folkloric use and validated bioactivities of this plant. The National Integrated Research Program on Medicinal Plant of the Institute of Herbal Medicine, UP Manila, have already developed a drug formulation from Olasimang Bato extract that is targeted to cure hyperuricemia and gout. So in this study, we wanted to assess the nephroprotective alongside the anti-clastogenicity potential of this plant to cisplatin-induced clastogenicity and kidney injury. So this figure summarizes the experimental workflow of this research. First, we gathered the plant, then air-dried it, and some of the fresh harvested sample were sent for identification and verification. So after air drying, we freshly prepared decoction with these concentrations prior to the in vivo assay. So for the in vivo assay, low dose of cisplatin was injected once a week for 4 weeks in mice, and plant decoction was administered 1 hour after cisplatin injection 3 times a week for 4 weeks. So this optimized animal model reflects the repeated low dosing of cisplatin used in the clinics, allowing us for detailed analysis of repair, recovery, and long-term kidney function. So after which, we extracted the blood for micronucleus tests, blood urea nitrogen, and serum creatinine evaluation, and kidneys were removed and subjected to histopathological and protein analysis. So this table summarizes the result of micronucleus tests. The frequencies of MNPCEs observed in mice treated with the plant decoction were statistically comparable to that of the negative control, hence it is non-clastogenic. On the other hand, it was found that there was a significant increase in the number of MNPCEs of mice treated with cisplatin alone compared to that of the negative control, confirming its clastogenicity. For anti-clastogenicity studies, a dose-dependent response was observed on the reduction of the number of MNPCEs compared to that of the positive control group, indicating that the decoction was able to mitigate the chromosome breaking effect of cisplatin during the treatment, and therefore it is anti-clastogenic. For nephrotoxicity studies, post-mortem gross evaluation revealed no abnormalities on all internal organs in mice treated with the plant decoction. On the other hand, severe intestine inflammation and pale kidneys were noticed in cisplatin-treated mice. The BUN and serum creatinine levels on mice treated with the plant decoction were comparable to that of the negative control groups, while cisplatin-treated group showed significant increase in the level on both renal biomarkers. Furthermore, the kidney sections from the negative control group and mice treated with plant decoction have normal renal histology. In contrast, CP-treated mice showed degeneration of Bowman space and capsule and tubular architecture, accompanied with fatty acid changes with cell swelling and possibly necrosis were observed. This suggests that the Peperomia pellucida decoction does not induce renal damage, hence non-nephrotoxic. For the nephroprotective assessment, the post-mortem gross evaluation interestingly revealed that the CP-treated mice who received 25 and 50 mg per kg of the decoction showed mild intestine inflammation. The administration of the plant decoction inhibited the increase of BUN and serum creatinine levels on cisplatin-treated mice. It was found that mice treated with 50 mg per kg of decoction have no significant difference with respect to that of the negative control on both renal biomarker. In addition, this experimental group showed the highest percent reduction of BUN and serum creatinine levels. The kidney sections treated with both cisplatin and plant decoction showed improved renal morphology compared to the cisplatin-treated mice. This further confirms that Peperomia pellucida decoction has nephroprotective activity against cisplatin-induced kidney injury. These bioactivities of the plant decoction could be attributed to the synergistic effects of the diuretic, analgesic, anti-inflammatory, and antioxidant compounds found on plant. In order to further elucidate the molecular mechanism of these observed bioactivities, we perform protein profiling on kidney tissue. The protein content of the experimental mice is summarized in this figure. The native paste gel profile of kidney obtained from all experimental mice revealed the presence of pro-apoptotic enzymes involved in cisplatin-induced nephrotoxicity, which ranges from 12 to 115 kilodalton. However, these protein bands in this region is hard to interpret due to overlapping. Thus, additional protein purification steps and experiments must be performed to further validate and quantify the presence of the pro-apoptotic enzymes. So we therefore conclude that Peperomia pellucida decoction is non-clastogenic and non-nephrotoxic and reveals anti-clastogenic and nephroprotective activities on cisplatin-induced clastogenicity and kidney injury in male albino mice. In order to ensure the safety and efficacy of Peperomia pellucida against cisplatin-induced clastogenicity and kidney injury, these are my recommendations. For further directions, it would be desirable to have a herbal drug preparation of this plant in treating clastogenicity and kidney injury during cisplatin treatment. So I would like to acknowledge this institution and people, and thank you very much for your attention.
Thank you. Now proceed to a question and answer. To our panel, we now entertain your questions to our presenter.
Okay, I can ask first. Hi, Mr. Lareno. Hi. What is the basis of your team for choosing the low, mid, and high doses, the 12.5, 25, and 50 milligram per kilogram dose? And then based from your results, you have determined the different properties like, and what can you say about the effect? Is it dose dependent, and what would be the most effective dose and why?
Okay, ma'am. To answer your first question, ma'am, we chose those concentrations by consulting the studies reported in the literature. So basically, for the nephroprotective studies, most of the ones reported in the literature use up to 100 mg per kg dose. So with that, we chose 12.5, 25, and then 50. And then to answer your second question, yes, there was a dose-dependent response in the anticlastogenicity and nephroprotective activity, and the results have shown that the dose 50 mg per kg was the most effective. So that's it, ma'am.
Is it significantly different from 525 and 12?
Yes, ma. Yes, it's significantly different po, ma'am. Because the ones treated with 12.5 and 25 mg per kg, the levels of serum creatinine and BUN was significant to that of the negative control. And while the 50 mg per kg, it was not significantly different to the negative control. Mm.
Okay, thank you.
You're welcome, ma'am.
Loreno, this is
Yes,
Dr.
ma'am.
Panganiban. Good evening.
Yes, ma'am. Good evening, ma'am.
Just few questions. You mentioned about the adverse effects of cisplatin. That's why you want to find out what are available in
Mm.
terms of preventing the possible nephrotoxic effects of cisplatin. Did you check as far as the literature as to how many of our cancer patients would still be receiving cisplatin for particular tumors? Because you showed the slide earlier, but there's no reference as to in terms of or the use or the utilization of cisplatin as still a anti-cancer drug, you know, for cancer treatment. Then second question is, what again are the safeguards that you took when you used cisplatin? Because as you mentioned, this is a highly toxic chemotherapeutic agent. So what were the safety precautions that you had to take while handling this particular drug?
So to
Okay.
answer, okay. So to answer your first question, ma'am, about
Mhm.
the percentage of like people having cisplatin
Using
chemotherapy,
cisplatin, yes.
yes, ma'am. I haven't checked, ma'am, but when I went to the uh, hospital, the MMRDC in Cagayan de Oro, which is the cancer center in Mindanao, the physician told me that they still like it's the number one option used in that cancer hospital. And
Mhm.
then so that's why ma'am, we chose cisplatin as the like subject of our research. And then for your second
And what's the
question,
incidence of adverse effect from cisplatin? Did you get from them the data that because they've been using cisplatin for their patients? What was the
uh,
incidence of nephrotoxicity from cisplatin in that institution?
in that institution, ma'am, I don't have the data. However, I consulted the literature.
Mm-hmm.
I just forgot to like put the reference, but those patients who are undergoing cisplatin therapy, almost 60% of them, 30 to 60% of them, uh, have the, uh, have got acute kidney injury during the therapy.
And the safety precautions you had to take
For the yes,
while handling cisplatin.
while handling the cisplatin, ma'am, of
What
course,
level of PPE did you use?
for the PPE, ma'am, I just use the normal like gloves, and then of course there's like goggles, and then wearing of mask during the experimentation. What is the main obstacle for this one is our like disposal of the mice. Mm.
Mm.
So and of course the chemical, I mean the use cisplatin.
Drug itself.
So that's it, ma'am.
And then you took the BUN and creatinine.
Mm.
Was it a ratio that you looked into, or did you look at a glomerular filtration rate, or in terms of using creatinine as a value and measuring in terms of reduction or
Mm.
normal creatinine or increased creatinine?
Mm. For that one, ma'am, for this one, we sent our sample to a diagnostic laboratory
Mm-hmm.
in the city, in Iligan City. And then we got the values. And then when I consulted the literature, ma'am, for long-term studies, the
Filtration
filtration
rate.
ratio is not somehow very reliable for long-term studies.
Okay, but this is
Mm.
an acute study, right?
This one, ma'am, is I don't
Yeah.
know if I can consider this as long term because we like have the study for 1 month, like every week. we injected cisplatin to the mice once.
So it's not
Mm.
chronic study yet.
Yes, 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 I suppose. Thank you, ma'am.
Okay, thank you.
Mm.
One quick question, Loreno.
For one last.
Yung pansit-pansitan, no? You had you had literature saying that it's anti-inflammatory, etc., etc. What made you think, or what was your basis for trying it on different direction, no? Parang pang cancer cells naman. Pang effect on the creatinine levels. Diretso ba ang mga literatures pointing to that?
For this one, ma'am, because we're very interested about the diuretic activities of the plant. So since cisplatin, along with most drugs, they are excreted via renal excretion. So basically, most of the waste products are filtered through kidney, or somehow they stayed or bioaccumulated in the kidney. So since uh, this plant has somehow diuretic effect, so we somehow think that maybe this plant is a good candidate for nephroprotective activity. That's it, ma'am.
Okay. Thank you.
Thank you very much, ma'am.
So if we don't have any more questions from our panel, we'd like to thank our presenter and our panel. Um, we would like to present the certificate to our presenters. The Institute of Herbal Medicine, National Institutes of Health, University of the Philippines, Manila presents the certificate of participation to our researchers for their presentation of their works during the third Philippine Herbal Medicine Summit with the theme Ensuring Quality of Herbal Medicines through Research, Regulation, and Cooperation held on January 25 to 27, 2021. May we request our panel and presenters to turn on their camera for a quick photo. To our participants, we will please be reminded that the evaluation form link will be posted in the chat box.
Our judges also. Hi, Ma'am Lynn. Hi, Ma'am Agnes. Ma'am Jessica, thank you so much for your time.
It's a pleasure. Anytime.
Quick photo with... Thank you po.
Thank you po.
Also, our presenters, are you all on? Saan yung iba? I think some of your professors are here also. They can also put on their cameras. Thank you.
Thank you.
For additional reminders, we would like also to thank our co-organizers, Flemex Vitex Negundo by Trevenod. We would also like to thank our sponsors, the Philippine Institute of Traditional and Alternative Healthcare, Binhi Biopharm Incorporated, New Market Link Pharmaceutical Corporation, and we would also like to thank UP Manila and the IMS UP Manila for the support and assistance. Again, we thank everyone for participating at the and we now close. This is the end of day 2 of the third Philippine Herbal Medicine Summit. Please answer the day 1 evaluation form available and clickable through the chat link. The link is posted in the chat box. Only those who have filled up the evaluation form will receive the certificate at the end of the summit. So again, thank you everyone and good evening.
Thank you, Paulo. Good night, everybody.
Thank you, Ma'am Jessica.
Thank you, ma'am.
Thank you rin. Ang dami pa namin participants until kahit gabi na. Thank you for staying on, everybody.
Congratulations.
Thank you, Ma'am Lynn.
Bye-bye. Night.
Mautang kami sa inyo.
Okay lang yan.
Ay, Sir Emil is here. Thank you po.
Thank you, Doc Cecil. Bye-bye po.
Thank you, Ma'am Agnes. Sa ulitin.
See you po.
Salamat po.
Bye-bye.